Kallio from Tampere University, Finland. Before I give a brief introduction about Professor, I would like to invite our coordinator, Dr. Kaushik Guha, for the virtual felicitation. Sir?
Yes. So Professor Passy, so because this FDP actually we are hosting through online mode, and we could not host all these speakers in our campus. So this is a standing invitation to you also. So you please, you are invited to physically meet us, our people, our department, our institutes, our labs, okay, in the physical mode when all the things will normalize after. So we hope that the country will be bounced back very soon. All the global world will be return to its normal situation very soon. So when the situation will come, obviously it would be my privilege to invite you to my campus. So for now, at this moment, we also take the privilege to felicitate you as per our Indian culture. So before your talk, so please accept our virtual felicitation. So thank you for accepting my invitation and really accepted to give one talk on very valuable topic today. So thank you, Professor Kallio, so for accepting our invitation and giving your time. Thank you, sir.
Thank you, sir. Thank you so much.
Thank you very.
Yes, sir.
Now, yeah, Anusha, continue.
Yes, sir. Professor Pasi Kallio is currently the head of Micro and Nanosystems Research Groups and the Vice Dean for Research in Faculty of Medicine and Health Technology at Tampere University. Since 2008, he had held a professorship of Automation Engineering in Biomicrosystems. Professor has authored more than 160 peer-reviewed articles, given several invitee and keynote talks in international conferences, has 15 patent applications, and has co-founded 3 spin-off companies. Professor Kallio's group has more than 20 years of expertise on microfabrication, microfluids, micro and biosensor and image-based measurements. Currently, the main application field includes cell and tissue engineering and micro-scale material testing. The group has developed several novel concepts for advised in vitro cell models, organ on chip. They include microfluidic, microphysiological cell cultivation environments with a strong focus on physio and hypoxic oxygen conditions. The conventional microphysiological cell has been demonstrated, for example, in neural disorder models such as multiple sclerosis and epilepsy and cancer research. The program has also developed several measurement solutions for microphysiological systems such as tailored microlights arrays, optical oxygen sensing, pH sensing, and biodegradable sensors. Professor Kallio is currently a principal investigator in 6 large research projects, including the Body on Chip Center of Excellence funded by Academy of Finland in 2018 to 2025. Furthermore, Professor Kallio is an coordinator of FiberNet in Innovative Training Network funded by MSCA of H2020 in 2017 to 2021 and PI in the FET Open Project Prime developing living cell systems for treating epilepsy. Now his talk is about microfluidic and microsensor for organ on chip applications. Thank you, sir. Please take your screen.
Thank you very much, first of all, for inviting me to this online workshop. And indeed, Dr. Guha, it would be then nice to meet also face-to-face at some point when the travel will be easier again. So thank you for the invitation. And maybe you can then advise me when my time is getting up. I think I have now about 50 minutes time because of misunderstanding, but maybe you can then indicate around that time so that I can start to wrap up. Yes, my talk today is about microfluidics and microsensors and their use in organ-on-a-chip applications. And what I thought that I will do, I will a little bit introduce our Center of Excellence here in Tampere about body-on-chip research. Then give an overview how microfluidics and sensor technologies are used in organ-on-chip applications, and I get and show examples from our research here in Finland. And then I will finalize my talk with showing some case studies that we are using how we are using these technologies with cardiomyocytes, so in heart applications, and with different neural cells. So before getting to the actual technology, so one slide why, and I think this is something that all of us are more or less aware that currently it takes long time, 10 to 15 years, to develop a new drug. And in average, the cost is more than 1 billion euro. So it's really expensive. And especially the clinical phases are costly. So those phases when the drugs are tested with patients. And the challenge is that the preclinical phases, so here, which are at the moment performed with animals and also in vitro cell models, mostly cancer-based cell models now. So they should be able to produce better candidates, so drug candidates that are more effective and safe for use, also that there are no side effects. And as mentioned, so these methods nowadays, so those are based on mostly 2D cancer cell models and laboratory animals. So what should we do? What would be an alternative to this? So how do we get more effective and safer drugs to the market faster and hopefully with smaller costs? And then okay, now sorry for this. So so the alternative is organ-on-chip technologies, and they are cell cultures which more and more in 3 dimensions combine various tissue engineering techniques and microtechnologies. And the idea is to try to mimic the way the tissue or part of an organ work. And the goal is not at the moment to build the entire living organ, but try to provide functionalities, so functional units of the tissue that have then the biological function that we want to have. And I think this may be the most famous example, originally from Boston, USA, and their lung chip, which is now provided by the company Emulate. And as you can see here, and as you maybe know, so these functionalities, different tissue and organ functionalities, are now studied in many organs. So I just listed here some. And we here in Tampere, as you will see, we will focus on heart and brain, and are building now also blood veins, so vasculature, to connect the tissues, and also look at the influence of the vasculature as an organ on other organs. But I will come back to that then in my examples. So our Center of Excellence here in Tampere, so I think I will maybe skip some of these, but this is from a starting point, a multidisciplinary research group. So we have 6 groups, and I think what I would stress in this slide, that we have 3 groups focusing only on biology, and then 3 groups focusing on engineering, and then we combine this expertise to develop these body-on-chip technologies and then in vitro models. And which expertise do we have here? So if we start from engineering, so as the cells are growing in 3D more and more, in biomaterials and hydrogel, so this is something that we definitely need in this organ-on-chip area. Then in addition to wet lab experiments, so we are looking at how we can make computer models. So Professor Hyttinen's team is making computer models of the cells. My group is making computer models on the environments where the cells grow. So my group, we are working with chips, chip design and production, microfluidics and sensors. And then we combine these expertise into the chip. I will today, as you remember from my title, I will focus on this part. And just very briefly from the stem cell biology side, so all the cells that we are using are based on human stem cells. So either adult stem cells, so to make vasculature and adipose tissue and bone, then iPS cell lines, so that we can get cardiomyocytes, but the same group is also working with hepatocytes, and then neuro group, which is then making different neuronal cells, and also looking at innervation, so how neuronal cells are connected, neurons are connected to different other cell types. Maybe this is I will skip. So kind of the big picture can be summarized here, that we want to make a patient on dish, so that we have some cells from the patients, for example, skin cells, that can be differentiated then to other cells, so that they have the genome of the patient. And then we have take those cells, differentiate them, and then we have this organ-on-chip platform, 3-dimensional cell cultivation environment, where we have biomaterials, instrumentation, measurements, and then the cells. And then when we have now this in place and we have the cells, so then we also aim to make a different trauma. For example, we are now looking at ischemia, how we can cause ischemia to the cells, or making mechanical damage or some other type of damage. Maybe give some drugs or chemicals to see their effects, and then to look at treatment. So how to treat then the diseases or trauma for using drugs. We can also study stem cell-based treatment or extracellular vehicles, any type of particle medication. And then the system should give us data, images, so that we can use this data and these images to make computational models and to understand the environment where the cells have been growing, and then the cells themselves. And by this, combining the knowledge and our observations, then eventually have a better medication and maybe stem cell treatments, understanding of diseases, and maybe at some point to support this to build human spare parts as well. So if you now look at then the technologies that we need, and now this is from Tampere University's perspective, so what we have been developing. So we need to have different technologies to stimulate the cells, to monitor the cells and the environment, and support then the growth of the cells. And then by combining these technologies, then we can develop these organ-on-chip models together with the cells. And the final aspect here, so what we are looking at as well, is then how to automate when we have this environment. So how do we make these experiments more automated? So the role of engineers. So I'm by myself engineer by training. So what is our role? So we want to make, as mentioned, the patient on chip, but we want also to have the kind of the healthy human. And we are then building this environment. So we have the cells there, we have biomaterials where the cells grow, we have cell culture medium, and then we would like to mimic this the body conditions. For example, that the temperature is correct or it changes. Then especially we have been looking at the oxygen concentration, that we can look at also low oxygen cases like ischemia and so on. So we aim to create the body-like conditions, either healthy, diseased, or damaged tissue, and then collect information about the environment and functionality using camera and different type of cells. And then based on this data, understand the particle mechanisms and then the functionality of our devices. And then about automation. So when I speak about automation, so what we are thinking, so we are thinking how to control the cell culture environment itself and how to control the cell functionalities. For example, how would we be able to control the differentiation of the cells to correct direction? And if you start with the cell culture environments, so why do we do that? So we think that in the current way to do cell cultivation and cell cultures, we do not necessarily really know what are the conditions where the cells grow. So they're in the incubators, but do we really know the incubator conditions precisely enough? And then when the cells are currently in these incubator boxes or incubators, so very often they are taken outside to either to do microscopy or make measurements, for example, measuring their electrical recordings. So then the environment changes when the cells are taken outside from the incubator to do the measurement. So the environment is not anymore the same as it is in the incubator. And also changing the environment in a controlled way is difficult. And then this leads to the fact that we don't really understand how the changes in the environment affect the cell behavior. For example, if the temperature changes, if the oxygen conditions change, and so on. And this is one example where we studied the changes of medium and temperature when the cells stay outside of the incubator for 30 minutes. So we take the cells, they are in the incubator, and then the pH there is this green one, 7.4, as it should be, and the temperature in the incubator is a bit more than 36 in the beginning. So these are the conditions how they should be. But when they are 30 minutes outside, so you can see how the temperature drops close to the room temperature to 26, and the medium starts to the pH starts to increase. So there's clearly there are changes in the cell environment when we take them out from the incubator. And the other thing is that here the door of the incubator was just opened for 5 minutes every 25 minutes, kind of mimicking the fact that people are taking the cells in and out. And you can see that there's also change in the pH and the temperature. So in the current way to do the cell research, the environmental conditions are not really stable for the cells, so there are changes. So this is kind of the vision that I have to develop this kind of technologies that would enable us to control the cell environment and also control the cell functionality, so that we have different technologies to stimulate the cells, either at single cell level or population level. Then we have the cell culture, and then we have measurements, either from one cell or single cell, and then we have population level measurements, so that we can stimulate the cells thermally, electrically, mechanically, for example, with flow or with stretching, and then with different biochemical factors, and then measure the responses of the cells, the thermal responses, electrical responses, and so on. So we make changes in the environment or keep them constant, and then we have measurements to collect data, and then provide this data to scientists so that they can, cell culture scientists and drug developers maybe, so that they can understand the cell and the cell environment better. But what we are also thinking now is that how we can use the same data to control these devices, these stimulators, so that we can direct the cells to the direction that we want, so that we can control also the functionalities of the cells. And for that, this is a busy slide and there are a lot of things. I will not go through them, but we have building these stimulation methods and measurement methods. So for example, we have micromanipulators to inject something into the cells. We have electrical actuators to change the electrical environment to stimulate the cells electrically. We have stretching systems. We have heaters so that we can change the temperature. We have means to control the oxygen, for example, so the gas environment. And then we have microfluidic perfusion channels so that we can have a flow there, and then provide actually also different, for example, drugs through the perfusion. And then we have patch clamp measurements. We have electrodes to measure the electrical activity. We have camera. So for us, camera is a very important sensor. So based on images, we can get a lot of information. We have biochemical sensors such as oxygen and temperature sensors. So, and now I'm giving examples how we are using these technologies then to study different cases with cardiomyocytes and neuronal cells. But before that, if you look at a little bit more about the functionality toolbox that we have, so we can make chips. So we have modular implementation for custom-made chips. So we can organize, and one thing that we have is kind of compartments, so that the cells can live in different compartments, and then the compartments are connected to each other. So we have different compartment topologies, and we have also different materials to build the devices. So here's one topology where we have the cells one after each other. Then we have another topology where and these are connected to each other, these compartments. Here we have this is another topology, so-called memo chip, which I will introduce later, and so on. So when you have means to make your own chips, so then you can change these topologies. You can make different kind of chips. Then the substrates we can change, so where the cells grow. We can guide the cell growth inside of these chips and restrict their growth. We should, and we do, monitor, change, and control the environment. We should have ways how to change the medium and supply compounds to the cells, measure the functionalities of the cells, and then integrate all these functionalities and connect them to the peripheral systems. For example, microelectrode array devices to measure the electrical activity, or pumps, and so on. So compartment topology. So always when you are making these chips, you should think, do I want to grow my cells in this study in wells, like here on the left, or in channels, as illustrated here on the right? Then do I put them on 2D next to each other, like here? So we have one compartment of cells here, here, and here, and then they are all connected to each other. And we found that this kind of system is very good for epilepsy. While here we have neurocompartment, cardiomyocyte compartment, and neurocompartment. So this we have found very useful to study the interaction of neurons through the axons with cardiomyocytes. So how the neurons innervate, connect the axons to the cardiomyocyte cells. And finally, here's a third example where we put the compartments on top of each other. So here we have one compartment which is filled with hydrogel. So these are living 3D in this compartment. Then we have a membrane, and then we have wells. So there's one type of cells living here, another type of cell living on top, and then we have a container for medium for this. So there's different ways to construct your chip. And the good thing is that if you have the fabrication capabilities, then you can fabricate these different compartments by yourself. So we are using different materials. So we are using PDMS, or actually PVP-coated PDMS in many applications. We are using also thermosets that we can 3D print, and we are using material called OSTE, so of stoichiometric thiolene, and then so basically for chip materials. So these are the chip materials that we are using, but there are of course other materials as well. For example, injectional moldable thermoplastics like polystyrene and polycarbonate and so on, which are very good materials for these applications as well. Then about substrate, substrates where the cells grow, we are using smooth, coated but coated surfaces. For example, cell culture plastic, glass, BDMS, and then for our sensor applications, titanium nitrate, silicon nitrate surfaces, or patterned. So here is one example why patterning is important for us. So we have here, we study innovation and develop the innovation. So how the actions of neurons connect to other cell types. So we have the same chip which you saw earlier, but now in this time we have neurons here, and we want to see how the actions go through, how they grow through from one compartment to another. And something that I will show in the coming slides, but there are tiny tunnels between these compartments, and only the actions can go through. So these are the kind of the actions are the neurons which carry the signals from one cell to another. So only them can go through. And if we have smooth surface here in this axonal compartment, so then at least here in Finland, I don't know how it would be in the southern hemisphere of the globe, all the actions turn to the right. So when they come out from one compartment, they start to turn to the right. Those, and same for here. So we can't really connect them, and they cannot go through. But when we made kind of grooves, tiny grooves, which we made using ATO, which is a polymer which can be light patterned. So they are following these grooves and nicely go through this compartment. So this one example where we are using patterned surfaces to guide the growth of the actions. But we can make this also from PDMS, we can make it from SU8 polymer, and even we have made used aligned nanocellulose from birch tree to use the nanofibrils of that material to guide the cell growth. And then also the substrate can be a stretchable substrate, so that we can cause their mechanical effects, mechanical stimulation. And always when we are using PDMS as a cell cultivation material, we need to code it. So PDMS is not so well supporting the cell growth that we would be able to use the native PDMS. So we have different extracellular matrices that we use for coding depending on the cell type. So as you can see again, so there are various options for the substrate substrates. So it can be made from different materials, but it can have also patterns, or it can be actuated so that it can be stretchable. Then to guide and restrict the growth, so as you saw before, so we need tunnels so that we can prevent the neural somas to grow from one compartment to another, or cells themselves, but only the processes. And we are using microtunnels. I think these tunnels that you can see here, maybe they are something like 5 micrometers in 5 to 10 micrometers in width, 3 micrometers in height, or 1 to 3 micrometers in height, and then several hundreds of micrometers in length. But microtunnels we need if we have a structure where we have the cells, the compartments on top of each other, we need to have then membranes there, and then different sizes of holes in the membrane, so that it's a porous membrane. So those holes work as a soma separator in these three-dimensional cases for us. So here you can see examples. For example, here there is an action thrown through the hole, and then its soma part where the nucleus is on the other side of the membrane. And then in some applications, we are using these kind of pillars or posts to separate the hydrogel, so that we can have hydrogels and cells on one compartment, which is here in the middle. And actually these black areas, what you can see here, these are medium flow channels where the medium is flowing. And we are using here these kind of posts or pillars to prevent the hydrogel, or to keep the hydrogels in the place where we want it to be. Okay, about controlling the environment. So oxygen is important parameter for us. So I will come back to this technology later again, but we are controlling the oxygen concentrations in these mini chambers that we can see here. In this solution, there's the same oxygen concentration in all of these 6, but we are now working on structures where we can compartmentalize the oxygen. So this is the device here. Then, so that we really know what is the oxygen inside, so we are measuring the oxygen concentration with sensors. So maybe making oxygen maps on the surface where the cells grow, but also now getting to the three-dimensional measurement, so that we can, in 3D cases, know the oxygen concentration also in not only at the bottom where at the bottom of the well, but also the other places where the cells grow. So getting from 2D to 3D in these measurements as well. And temperature. So temperature is something that is important to keep, or sometimes to change. So we should have means to do that and measure it also. This is one example also coming again later. And then medium change, so either manually in open top wells, as you can see here, or then with the microfluidic pumps in channel-based systems, like here. And then of course, then there's again a lot of possibilities for pumping, and I will not go through all of those pump types, but there are many possibilities for that. About cell measurements, so we have many electrically active cells, so neurons and cardiomyocytes, both of them. So we are using microelectrode arrays. So these are tiny electrodes placed into the cell area, and they measure the electrical activity. So these dots here, which you can see, so these are the electrodes. Here are cardiomyocyte clusters, and then we get the signal, electrical signal from the cells through these wires to the pads, where we connect then the actual measurement amplifier, and then we can get signals like this. And we are looking at different configurations of how to place these electrodes. So in different chips, they need to be placed in different, so that we can measure the activity in different compartments. So we have to be able to change the configuration. We are looking at different electrode materials and also insulator materials. So it's also important that we can really separate or insulate the wires, for example, from the medium and from the cells, that there's no disturbance through that. And then, as I mentioned earlier, images are a very important tool for us. So here is user interface of our software that we made for looking at the cardiomyocytes and their beating. So from that image signal or the videos, we can get a lot of information about the behavior and changes of the cells, as I show in the cardiomyocyte case. And then combination of these, of course. And then integration. How to integrate these functionalities so that they don't influence the other functionalities, and how do we make the interface to the peripherals like amplifiers, microscopes, pumps, gas bottles, and so on. And you saw many different functionalities and technologies. And by no means we are there that we would be able to put all of these together. So we are able to have some combinations. Some of these can be integrated at the moment, but not all that you saw in the previous slide. So this is one of the future works and challenges, I think, how we can efficiently do this integration. Then I have those chips here. I will just show that we have many and then don't use more time now. Let's come to the questions then at the end. I see some question in the chat, and it's a bit difficult to read and answer right now, so I will come back to those then later. So a little bit about the case studies. We have about 15 to 20 minutes. So I think we have time to look at them at least briefly. So if we start with the cardiomyocyte, and this is just a slide to remind you that I think that one team with one type of expertise cannot do all the work that you saw before and do the integration and do the cell work. And that's why we have a combination of teams working on this. So it's of course a bigger group than one team of mine. So Professor Aalto-Setälä, she is expert in cardiomyocytes, and she is doing cardiology in clinics by herself as well. So she knows the patients and she knows the cells. Then Jukka Lekkala has been developing the sensors, electrodes and oxygen sensors. She retired, and his team is now under my team. And then Jari Hyttinen, who has been then using the data that we collect to make in silico models of cardiomyocytes, so that there are computer models, better and better computer models of the cardiomyocyte functionalities. And then also doing different imaging technologies, or developing the imaging technologies. So we have made this kind of model cell culture environment with an idea that we would be able to combine as many of these functionalities that I have shown to you together in a modular way. And here is an example how we have been using it for the cardiomyocyte study. So we have here a microscope, which is actually self-made, so that it can image either from top or from below. Then we have oxygen sensor set up now in this version here below, so we measure the oxygen concentration at the well. Then we have microelectrode array, so that we can measure the electrical activity. And as we have the camera, so we can use then image-based methods to analyze the beating. And then we have heating and temperature measurement here as well. And from this video as an example, so when the cardiomyocytes beat, so we can analyze the beating rate, and we can get data like here. And from these variations in the beating rate, we can see a lot of we can learn a lot about the cells and then how they respond environmental changes, or how do they respond to drugs, for example. And this is one example of the temperature response of the cardiomyocytes, so how the cardiomyocytes respond to the temperature changes. So this is, let's play it again. This is the normal temperature, 37 of normal temperature of the body, and of course, as we can see that what happens when we reduce the temperature, the beating rate reduces, and you saw how radically it changed. And of course, it's an example also that if we would have these cells in an incubator, and then we would move them to the microscope to do imaging, for example, so clearly they respond to that. So in 25, the beating rate is totally different. And here we have then some idea what we can get from this, that we have a temperature that has been cycled. So we have studied beating frequency, how many beats per minute from this image, relaxation time of the cluster, contraction time of the cluster, amplitude of the contraction, and so on. So we can get a lot of information and data from these images, which can then be used for different studies. And here we use it to study the temperature response. And the same thing for oxygen. So here we have cultivated the cells in different oxygen concentrations, and there is again clear indication that the oxygen concentration has an effect, and the lower it is, the slower naturally the heart or the cardiomyocytes beat. And from this we can then again get a lot of data in the same way as the temperature example. And we can also do cycling, so we can make the condition hypoxic and then do reoxygenation again and look at also this recovery phase. And the technology is needed for this. So what did we need to integrate to collect the data? So we had to have the gas supply system, this ring here, to change the oxygen concentration. We had oxygen measurement there. We had the microscope with camera, and then the image analysis and heating and temperature measurement. So with this, actually quite many things we needed to integrate. And microscopy. So this I already explained, but from this little bit blurry video, you can see here from recorded video, we can calculate the velocity vectors in this, and from the velocity vectors, it's possible then to calculate the beating signal from different areas of the cell. So we can also look at either the cell or the population, and how if there's any differences in different parts. Then so we can see that there's a kind of a type, healthy type or healthy population as a control population. We can calculate a lot of parameters, as you can see here. How long is the contraction? When does it end and start? How long is the relaxation? And so on. And then compare it to diseased population. So this is a cell type or cell population that has been obtained from a patient that has a genetic heart disease. So the beating is different. The shape of the curve is different, and those can be then analyzed. But oxygen measurement, so we use here luminescence-based oxygen measurement, and how it works in our case is that we have cell cultivation substrate, for example, glass, and on top of that we put the polystyrene, a very thin layer of polystyrene, and in this polystyrene layer we have fluorescent dyes that are sensitive to oxygen. So when the oxygen concentration changes, then the fluorescent properties of the dyes change, and we are looking at the either the intensity, or then how long does it take that the intensity changes. So we excite the dyes with certain wavelength and then measure the fluorescent emission, so the fluorescent signal with another wavelength, and use then this for getting information about the oxygen concentration in the chamber. And here also the biocompatibility and cell compatibility issues are important. So we had to do quite extensive work so that we were able to keep these dyes inside a polystyrene matrix so that they didn't leak to the cell culture and harm the cells. So we developed the covalent bonding method to keep the dyes inside the polystyrene. And now we have shown that more than 14 days, more than 2 weeks, there's basically no influence now on the dyes to the cells. About the gas supply, this is just a reminder that, of course, in human oxygen concentrations are totally different than in air, in ambient air. And the challenge in a way at the moment is that in standard cell cultures, the oxygen concentration is 21. And in tissues, it's between 2 and 9 inside the body. So for example, in brain, it should be 4 something, in kidneys, 9.5 something, and so on. So we have been starting to look at how can we control and change these oxygen concentrations in our chips if needed. And this is one way to do this currently. Of course, there are technologies how to culture the cells. For example, in hypoxia, if you want to study really low condition oxygen concentration. So you put the cells inside of this box where there's the desired low oxygen concentration, and then you can work through these kind of gloves so that you can keep the oxygen concentration as you wish. But we wanted to move this to the chip format and make this kind of portable hypoxia system. So now the cells are growing here in this box, and for all of these cells now the concentration is the same. And then you can take this part, this box, and put it under microscope. And there's a heater, there's a connection to the gas supply, a small bottle that you can see here inside the box. It's transparent, so you can do the imaging. And this has a battery, so it can last several hours. So we made this kind of portable incubator where you can have the desired oxygen concentration for long periods of time, and you can also do, for example, microscopy or other measurements without disturbing the cell environment. And we had a demo already 4 years ago in our exhibition hall here, and we showed that we can keep these cells alive. They were feeding cardiomyocytes 3 days in this conference exhibition hall, which is not a research lab at all, but they were growing here in these chambers and in this portable incubator for that period of time very easily. And later we have shown that they stay alive several weeks. I think I will now skip a little bit this so that we can move on. Maybe just mention that we commercialized this, or a member from my team commercialized this by Genuine Microsystems company. It's provided through Baker and also now through Leica as well in one of their applications. This is a kind of a cryo application. Heating, I think I also skipped this so that I have time to show a little bit of the neuro part. But basically, what we have been developing is that how to measure the temperature so that we don't need to put sensors inside the place where the cells grow. As we saw, we have already the electrodes there, but if we start to put other sensors, so it might be difficult. So we decided to look at the methods that we have the sensor outside, and then we make a model, numerical model, to estimate what is the temperature inside. So, and then we used a PI controller to control the temperature also if the outside temperature was changing, and we got pretty good results. So now here you can see the difference between the set temperature and the measured temperature when the sensor is not inside our compartment where the cells grow, but it's placed outside. So we can estimate it pretty well with plus minus 0.3 degree precision over 4 days. So now we don't need to put our temperature sensors inside, but they can be placed outside just next to the chamber, and we can control and also regulate the temperature as we wish. We have been also looking at cell stimulation by stretching. This is also something that I will now a little bit speed up with this. I will maybe just show how it works. So we have 2 compartments and an air channel in between. So the cells grow inside on this kind of stretchable PDMS membrane. Then we put vacuum here outside, and when we have vacuum in this chamber, so then the inner ring, it bends like this, and when it bends, it stretches the membrane, and we can change the amplitude of the pressure, the frequency of this pressure, and then we can have different different stimulation patterns here. And the good thing with this method compared to the others is that we have very good imaging capabilities. So we can get good transmitted light microscope images and confocal microscope images, fluorescent images, and so on. And where we have used it, so we have used it in cardiomyocyte study. So if you don't stretch, if the cardiomyocytes are growing on heart plastic, for example, so then the sarcomers, which are responsible for the stretching of the cardiomyocytes, so they become random like this. They are not aligned at all. But when we stretch, then we get this kind of aligned sarcomers that are much closer to the real heart cell, the real cardiomyocyte. And now we have this device in multiple format as well, and you can hear the sound of this stretching system. Maybe I will go a little bit forward. You can see then the stretching, and here's the device that we built for controlling these systems. And then take the last look at, and then as you can see here, we have 6 devices as well. So this diameter is changing, so we can then stretch the cells. Okay, and then last examples about brain. So here our collaborator was Dr. Susanna Narkilahti, and I will show this example of multiple sclerosis. So we made a chip where if you think of multiple sclerosis, myelination, lack of myelination is one problem there, and it means that when we have here now the cell bodies, the neurons, and the axons. So the axons in a healthy brain are covered by myelin, and myelin is kind of an insulator. So in the same way as in electric fire, there's an insulator, plastic insulator. This myelin works in the same way, and in MS disease it starts to disappear. And when there's no insulator in the electric wire, so there are electrical problems, and these can be seen, these electrical problems, as some symptoms as seen in multiple sclerosis. So and this myelin is created or made by oligodendrocytes, another cell type that lives in the brain. And we wanted to make this kind of model where we have neurons again on 2 compartments, then another compartment where we have the axons, and where the oligodendrocytes have access so that they can do the myelination. And we wanted to study if we can do this with the human cell types. And maybe to make the story short, I can say it's not that we were not able to do it with the human cells, but with rat cells we made it to work. Not exactly like this, but by placing the oligodendrocytes here, so we saw the myelination, and it can be seen here in these images by a skillful eye. But I will not explain it now in further details. But this is an example, several compartments, 2 different neuronal cells, and the idea is to look at MS disease by looking at the myelination of the actions by the oligodendrocytes. I will skip this. Here, just to mention that we are using different configurations. Here we have the serial and then more parallel configuration to study axonal damage disease, epilepsy, and stroke. And then we have this kind of the lid, so to keep the oxygen concentrations as we saw change it, and then microelectrode arrays below, and then do the electrical recording. And I think this is now maybe here where I will stop. So I will not go through all the technologies that we needed for this, but maybe to stop here. And if you have any questions, so please, maybe there's now time for a couple of questions, and I will just go to the end to thank you for your attention and invitation once more. And we
Thank
can.
you. Thank you. Thank you, Professor. So fantastic presentation, fantastic work. Actually, in fact, we have seen that you and your group is doing some very high specialized and advanced level research work is going on at your end. So it would be very honor and privilege for us also if we can collaborate with you and contribute something and can work under your guidance or under your group. So that will be our actually the motivation and the intimation. And we want that also all the participants will also express their feelings and willingness to collaborate with you, to work with your team. So jointly India and Finland and your university and our university can do something better and contribute something on this domain. So thank you very much. Now the panel is open for some interaction, maybe I could say, not a question answer, it is an interaction session. So the house is open for all the participants to ask few questions because we are running out of the time. So we cannot take all the questions. So only few questions, okay, we can ask and to professor. So the professor will be very happy to reply.
Yes, indeed. And I can also then, if there are questions in the chat, I can also answer the questions in the chat when there's the next presentation.
Yes. Okay, you can do this.
Should I read out, sir?
Yeah. Anusha, you can read out. Huh?
Okay, sir. Okay, fine. So the first question, how to plot graph from the electrode detection data? In which software you people are using, Professor?
So we collect the data. There's a special software that is functioning and working with the hardware, with the amplifier. But then when we have stored the data, so we typically use MATLAB to do the analysis. So analyze the signals and then plot the signals. So we use MATLAB.
Good morning, sir. I'm Satish. I would like to ask a few questions, sir.
Good.
Sir, you were talking about electrical stimulation of the cells.
Yes.
So at the time of culturing and all. Sir, I have a question. So electrical stimulation would be a harsh one to apply for the cells. So I want to know what is the maximum voltage till now you have applied and why so?
I have to look at now. I don't remember it by heart. So we are not typically using electrical stimulation. It's something that is not the standard way. However, we had collaboration where we had primary cardiomyocytes. They were from rat, I believe, and they do not beat without electrical stimulation. So in order to make them to beat, so we need electrical stimulation. But now I have to say that I'm sorry, but I don't remember by heart what was the voltage that was used in this study. Some millivolts, but I don't remember now exactly. And I think it's also, I would say that it depends then on the cell type what you need to use. These cardiomyocytes that we are using, that you saw, they start to beat spontaneously. Here we don't need electrical stimulator to keep the beat. But for primary cells that are from heart, from animals, so they need to get the beat. Otherwise, they don't beat. So they need to get the rhythm, the pacemaker, in a way.
Okay. Sir, another question. So we have electrical properties, as you know that every cell has mechanical, electrical, chemical, and all kinds of properties if we go down to the cell level. So the question is if we could study a single cell, the electrical properties of a single cell, is it possible for application
Satish, your voice is breaking.
It's quite difficult now to get the question.
Yes, sir. Yes, sir. I can talk to you, sir. Sir, is it possible if we could study the electrical property of the cell? Is it possible to make a personalized diagnostic device such that we can avoid this usage itself?
I heard that is it possible to separate the cell, but is it possible to write it in the chat, the question? Maybe it's easier to answer.
Yeah, Professor Kwasi, is it audible to you?
Yes, sir.
I had posted a question regarding making a composite of chitosan and functionalized graphenes. So I was going through your profile and I found that you are a part of the FiberNet project which works on biocomposites.
Yes, we studied chitosan together with the nanocellulose. So we have been using nanocellulose films.
Okay.
And the nanocellulose itself, we were not able to make it really mechanically durable. And our experience with chitosan and nanocellulose is that yes, we can make.
Yeah. So like ours is a R&D center of DST Government of India. So if you want to collaborate in your team, like through some collaborative work, so it will be possible at your end if you submit a proposal for a bilateral work.
Yes, I think we can maybe discuss then separately.
Yeah, sure.
And the details and what would be then.
I'll write to you.
Very good.
Okay.
Good.
Okay. Thank you so much.
Thank you.
So another question that Satish has typed actually. So if we could study a cell electrical properties, is it possible to generate personalized diagnostic devices?
Is this now referring to? So do you mean personalized devices? I didn't now get the question.
If we could study a cell electrical properties,
yes,
is it possible to generate personalized diagnostic devices?
So we are studying in vitro, so outside of the body, and I think so. What we can do is that we can take skin cells, let's say from me, and we can differentiate those skin cells to cardiomyocytes, and we can then study kind of the normal beating of those in a lab, and then if we measure these properties, so can we convert this understanding somehow to a personalized device that would then measure my especially my heart behavior, as I believe that this means in vivo. I think there's a kind of a long path. So what we want to do, what we aim to do, is then when we have, let's say, my heart cells beating there in the laboratory. So we want to see how this population responses to different drugs, for example. So to have personalized drug treatments. So this is something, and in a way, personalized diagnostics through that. So if I don't know if I totally got the question, but what we can do in the future, somewhere in the future, is to look at the personalized treatments, personalized drugs, and also if there are side effects of drugs to the heart, personalized, because we can get these cell cultures from the individuals. We can differentiate different cell types, and then these cells have the genes and genome of the patient. So personalized treatments, yes.
Thank you, sir. Thank you very much. I have few other questions I will write to you, sir. Thank you. Thank you very much.
Okay, thank you.
Sir, another question, just only one question. Okay, we can take it. So, what type of microscope you are using real-time monitoring?
We have built our own microscope as well. So it's an optical microscope that has fluorescence options. So we are using bright field. Either we have the objective from bottom or from the top, and then we have fluorescence option to see fluorescence signal as well. And then we are using just a lot of commercial microscopes from Leica, from Zeiss. So I think there's no one single microscope that we can use. I think then the camera is important. So what kind of camera you have, so to collect the images. So, and of course there are also many options. So not a single camera or not a single microscope works, but I would say that then the challenge is that why we have built our own imaging systems is that we can more flexibly integrate our chips and other sensors, oxygen sensors, and these electrodes. It's easier to integrate all of this together when we build kind of our own imaging system, and that's why we have done it. But basically, when you have chips, let's say also commercial chips, you can use almost any microscope with those. But then you end up maybe with challenges of integrating other functionalities that you want to have. But there's no I would say that, yeah, many microscopes can be used.
Yes. So the last question of this session, please, Dr. S. Senthil Kumar. So you have raised your hand. You can unmute yourself and can ask the professor the last question of this session. You can take. Sir, please, Dr. S. Senthil Kumar. Dr. S. Senthil Kumar, are you going to ask some questions? Okay, sir. So I think there might be some problem. So thank you once again for answering all the queries and giving your time. So it would be our privilege that, okay, you have joined and you have accepted our invitation to deliver a talk on this forum. All the participants, I think, immensely benefited, taken so many good informations on this field, and obviously they will be motivated to pursue the research on this field. So whoever, if you want to connect with Professor, you can connect to me, or you can send me mail, or also you can directly send a mail to Professor Pasi Kellio. So for your further collaboration or further communication. Thank you very much. So now, Manu sir, we can conclude the session.
Yes, sir. Thank you, sir. Thank you for your valuable information about this drug development, control of cell culture environments, overview of cell culture substrates. It's a very informative program, sir. Thanks for accepting our invitation. Thank you so much, sir. Have a good day, sir. Thank you.
Thank you. And sorry making you waiting. I'm really sorry for that.
No problem.
But thank you
sir.
for the invitation.
Thank you.
No, sir.
Thank you.
So it was too early at your end that I know. Now it is, I think, maybe at 8 o'clock or 8:30.
9.
Yeah, 9 o'clock maybe.
Yes.
At your place.
Yes.
Thank you.
Okay, thank you.
Professor.
Thank you.
Thank you, sir. Thank you so much. So next session.
Yeah, so the next session will be started, I think our honorable speaker, Professor S. Raghavan sir, has already joined. So
if you want, you can take 5 minutes.
okay, I think Preeti, we can start now, huh?
Sir, okay, sir. Sir, is my voice audible and clear?
Achha, your voice is low, Preeti.
Is it audible and clear, sir?
Yes, now.
Okay. Welcome back to the second session of the day 4. It is really encouraging to see you all enthusiastic in this session. This morning amidst us is a most experienced professor and a great researcher, Professor S. Raghavan from NIT Trichy. Sir, on behalf of the Department of EC and NIT Silchar, I extend to you a warm welcome, sir. I would like to introduce Professor S. Raghavan to you all with a brief biodata. Dr. S. Raghavan, Professor in Electronics and Communication Engineering Department, National Institute of Technology, Trichy, has 35 years of teaching and research experience. His interests include microwave integrated circuits, RF MEMS, BioMEMS, metamaterial, and microwave engineering. As a proud research scholar of Professor Bharati Bhatt and Professor S.K. Kaul, CARE, IIT Delhi, he has established state-of-art microwave integrated circuit and microwave laboratory in NIT Trichy with the help of Government of India funding. He won Best Teacher Award twice and conferred with Honorary Fellowship of Ancient Sciences and Archaeological Society of India. His first research scholar obtained a gold medal for the best thesis of the year. He was a short time visiting fellow in California State University, Northridge, USA, and was also awarded to conduct tutorial in APEMC 2010, Beijing, China. He is an organizing chair of Indian Antenna Week 2014, Chandigarh, and invited to be a session chair in PIERS 2013 Symposium, Taipei, Taiwan. He has to his credit 123 research papers in national and international journals, 94 in IEEE Explore, 132 international conferences, 28 national conferences. He also filed 3 patents and guided 16 PhD scholars. He is a senior member in more than 20 international and national professional societies, including IEEE, BAS, IEI, IATE, CSI, TSI, ISTE, ISSS, NPC, ISOI, ILA, IELTS, and ASI. He organized various workshops of national importance. Professor S. Raghavan has contributed a lot in the development of state-of-art library and hospital in NIT Trichy. He changed the clinical dispensary into a 24-hour hospital as the then chairman of the Hospital Modernization Committee. He is an executive member of TSI and also in the Tamil Nadu chapter of TSI. He is also the founding faculty advisor of IEEE, MTT, and APS student branch chapters NIT Trichy. He was the president of REC Trichy Faculty Association for 2 years. Professor S. Raghavan is the author of Microwave Integrated Circuit Components Design through MATLAB, and he made a great impact in microwave engineering education among the student community at large. Sir, this morning we are very privileged to have you amidst us, sir. I invite you for the talk, and the stage is yours.
Can you hear me?
Yes, sir, clearly.
Oh, dear valuable participants, I will tell you why I call valuable. Interdisciplinary young person, Dr. Guha, professional master of ceremony. I do not know whether to call madam or doctor or professor. Anyway, I know she's a scholar, Preeti.
So thank you, sir.
Yeah. Other team members, Shaurav, then Satish. Satish, I find him asking very good questions. And I have got a lot of regard for all the participants. Now I know every one of you. I know some of the people are already established, like Dr. Srinivasa Rao also is there. I don't know whether he's continuing. Many more persons. By the way of your chat, I could, it's all interesting sentences you are telling. So with all that familiarity, I am entering this session. And this will be little bit different also. First of all, BioMEMS is a multidisciplinary topic. Each one will talk in a different way. Many people should join, and then only this BioMEMS will be the most successful one. So that way, and also during in the afternoon time, I will not tax you much. I will not go very deep in all those biological. I'm also not good in that. In a way that I will definitely make a little exposure to certain things, at least in something which you are not aware of it. So that is how my talk will be. So that's why today morning only I added that 3 words and overview. So with that, I start. Before that, let me ask one small question just to know which I should talk more, which I should not talk more, and also not to test any one of you. Can anyone tell, mute or put it in chat box so that team members will tell me who is said to be the father of, you know very well, our Calcutta person yesterday talked wonderfully, the nephrologist. Another Calcutta person is said to be father of radio, millimeter waves, many things. Now my question is, who is said to be the father of plastic surgery? Because many things, Indians stand first. Space application, what Mars mission could achieve, no one did achieve in the world. Many things, we are number one in the world, including biomedical. Biomedical, we are number one in many aspects. I will tell you why. So that way, just want to know whether anyone knows who is the father of plastic surgery.
It's Shushurta, sir.
Very nice. Who is telling?
Prasanna, sir.
Prasanna, okay. Anyhow, he may know that plastic surgery person is Shushruta, but I don't think he will know there's a statue for him. There is a very big statue for Shushruta. That is in the God's own country, in Trivandrum. There are many statues throughout India, but that's a statue for this stalwart who is very much connected with the BioMEMS. Shushruta, he only laid the way for plastic surgery throughout the world, not for India. Likewise, we can say Ayurveda also. Like that, many things. I will tell you one small information. May not be an authentic, but it is authentic. We cannot say official one. That way, we are number one in medical tourism also. And certain things that is solved, we will be definitely be called authoritatively number one in biomedical and medical care. So Shushruta has played a very big role in the plastic surgery, where there was no much on anesthesia, many things. Likewise, people should know what Ayurveda also has done. So similarly, we can also do wonders. People are doing wonders. I will tell a little bit away. Don't think that I'm not talking the subject. Some 60 years before, I came across one Guha, not Kaushik Guha. That person, even now, is there. He's, can anyone tell whom I'm referring? He's the only person in India who has completed BTech, MTech, PhD in engineering, then MBBS and MD. That Guha, can anyone tell? In fact, he was an honorary distinguished professor along with our speaker, Roy, first afternoon day image processing, molecular image. He was the head of the department. Under him, that stalwart was working. Can anyone tell? Why I am asking is, if you know the history, that will make you become like that. Can this Guha, Kaushik Guha, tell who that Guha is? Yes, that person's name is S.K. Guha. And he was head of the department in biomedical engineering in IIT Delhi. That is the best biomedical engineering department in India. Next to that is IIT Bombay. Then IIT Kharagpur also. The person who gave a talk on the first day afternoon, he was only a microprocessor person. Then he knew that time means what, 30-40 years before, he knew much on image processing. Then he became this much knowledgeable only because he was moving with all super specialists and interdisciplinarian medical people, biomedical people, and he became the head of the department of the School of Medical School Technology in Kharagpur. So these are the institutes where they give. I think NIT Silchar also has started doing. They take MBBS people, MD people, then give them doctorate. By that, many things can be achieved. So with that, I want to make a start. Definitely, engineering and medical should join, and the miracles will happen as is happening in the last 2 years. Necessity is the mother of invention. That is how many inventions started, even during this COVID time, because COVID has to be cured. So that's how in a very short time, in software and hardware, many gadgets came. Oh, people keep on talking on AI and this machine deep learning and many things. All these things have combined. That's how they could easily find out, even in a big crowd, they can easily discriminate who are affected by COVID. It is not only for diagnosis, for therapeutics also, our technology is being used. Only thing is acceptance rate is not so quick. It takes a little more time. And that's what everyone is seeing in COVID also. You can even say BioMEMS, some of the speakers also told. The only thing is they did not mention it as a BioMEMS, but they talked the application of BioMEMS in now the dreaded, the most the disease which the whole world is afraid of is the COVID only. So even the COVID can be cured by BioMEMS. Certain cases we don't tell that as a name. We see a peacock, but nobody knows. Very few only know it has got nanotechnology. And nanotechnology also play a very big role. In fact, the definition of BioMEMS is it is a platform where nanotechnology will start working. So with that, I will go to the subject. And 4 hospitals, 4 colleges, institutions I mentioned, it is all shining in biomedical only because each is associated with some medical college, very good medical college. Delhi, IIT Delhi is associated with the all Indians for medical science. Likewise, in Chandigarh, it is being associated with the PGI. Likewise, JIPMER, like that, many are getting association with the big hospital. Then only one can thrive. That's why I understand this NIT Silchar also is getting associated with that Silchar Medical College, and also I come to know recently about the cancer hospital. And through these lectures, I come to know some of their association with the Calcutta hospitals, many more in Singapore, and some of the MEMS persons in IISc. With that only, and I know very well, everyone knows the Center of Advanced Study in Biomedical Engineering in India is again situated in the God's own country, Sri Chitra Tirunal Biomedical Center. From there also, I think you have one speaker. So that center, being the founder director, is the son-in-law of a king, Chitra Tirunal. When you call him, he will come along with a small heart. Everything, artificial organs, it's all known for that. You can see it physically, nothing like imagination. Such a wonderful center is SCT. So like that, if anyone is associated, engineering institution is associated with this medical college, I understand that KLE where Dr. Rao is also there, he is also associated with the hospital. Unless otherwise you are getting associated with a hospital, top-notch hospital, your work will be just for research publication, papers only, PhD. It won't be of societal value. Now I understand in these last 4 days, many would have come across the people whose research outcome is directly usable for the societal value. That is how the research should go. And biomedical engineering and BioMEMS is the best subject that can be of much value for the society. And now what you are seeing is not simply a chart placed over the cake. You go to Yahoo or Google, just type Smith chart. This will be coming as the first picture whole world can see. This is the culmination of a postgraduate student. What he did was, he has done on a JPG plot. That is how he created. Why am I showing this to you? Because this is needed for the design of many of the RF MEMS circuit. We have got BioMEMS antenna. We say implantable devices. So in the implantable devices, if you want to signal, you should have the BioMEMS antenna. Likewise, many of the devices can be designed by this small chart. This chart, ECE people will be knowing, is a Smith chart. And again, if you want to know, go and see the website of P.H. Smith 1938. You'll come to know many more things. Whatever you want to tell about the electrical engineering, electromagnetic, or bioelectromagnetics, you will come to know with this chart, with a small geometry box, you can easily design the structure. Design the structure means what? Length and the width only. Take a three-dimensional one. The third dimension is the manufacturer's concern. The length and width only you have to design. That should be, that is not in centimeter or millimeter. Now once you go to the MEMS, it is all in micrometer, it's all in nanometer. But once you tell in terms of lambda, it is same in any frequency and for any application. So many things can be designed with this simple chart. No need to copy from any of the papers. And now he is the person, because of him only we keep talking MEMS, RF MEMS, BioMEMS. And there are many sentences told by many people. That single sentence made a big history. You know, Moore's law. Like that, he told there's plenty of room at the bottom. That sentence made many come for the new technology and integration of multidisciplinary. It is not only engineering different branches. It should definitely get amalgamated with the multi-speciality of the medical side also. That's how the first MEMS started, micro electromechanical system. I hope you know the first book by Mark Madhu, especially in all these kind of things, more than engineering, medical, the core physics people will do better. That's why if you see the last ISRO chairman is a physics person. Likewise, Mark Madhu, some top people, initial people, they're all physics people. They have done wonders. So like that, there are some, I will show you some Bibles which can be referred to master BioMEMS activity. In any case, this sentence fetched him Nobel Prize. And two things your master of ceremony told, many things about two things, about myself involved in the microwaves, millimeter, like that. Anything, any frequency more than gigahertz, you call it as a microwave. Then little more than 30 gigahertz, you say millimeter wave. So these are all, see, this micro electromechanical system, you plan it for the RF frequency. Then it is called RF MEMS. It is not MEMS at RF frequency. It is planning and fabricated and tested for RF frequency. Both are having difference. And that is the second stage of MEMS, that you call it as a RF MEMS. Many of the technology of design of the RF MEMS is very much used in the BioMEMS. The application of RF MEMS in the biomedical application and also a little bit of MEMS, you call it as a BioMEMS. And it is also, that's why if you talk in terms of lambda, it's a universal value. But if you talk in terms of size, here we call it as a centimeter, or you can call it as a millimeter. In BioMEMS, everything will be micrometer or nanometer. And as far as the planar view is concerned, RF MEMS, BioMEMS will be same. Whereas the side only you can see the difference between RF MEMS and BioMEMS. See, you know very well, if you take air, air is a lossless material because epsilon r is equal to 1. Epsilon r play a very big role in biological effect of microwaves. See, if you take a year, the epsilon r is equal to 50. Where is 50 and where is 1? That is why when you keep talking with the cell phone, the ear lobe will become reddish. You get a heating effect. Ear will not get heated, but you can see the reddish figure. In any case, long usage of lengthy usage of cell phone is also not good. Accordingly, one should not use more than 6 minutes per second. Anyway, we'll see it little later. In any case, what I want to tell is you want substrate because miniaturization. Because of miniaturization, if you use substrate, there are some substrate. You may ask, what is the substrate for implantable antenna? What is the substrate for some organ? So that's what we call it as a substrate. I can proudly say one Indian from the God's own country by name M.T. Sebastian, his book is the best book for about to know everything about substrate. And incidentally, I can say there's a Bible given by an Indian by name O.P. Gandhi. His book on biological effect of microwaves play a very big role. In any case, dielectric constant is very much essential. You should know what is the epsilon r of each part of a body. Then only you can say what is the interaction. Just for comparison, I have told epsilon r is equal to 1 for air, 50 is for ear. So one should not keep playing with cell phone for a longevity. So if you want to have the benefit of air and also the substrate, you can combine both. So that is the principle of BioMEMS component. You remove the substrate. If you remove the substrate, air will be there, substrate will be there. Substrate is for miniaturization. Air is for reducing the losses. So that way, the layman's sentence is remove the substrate. But mechanically you should do it, methodically you should do. So that's why this 54.7. 54.7, how it comes is you take any BioMEMS component, if you take a device, it will definitely the horizontal, first of all, you cannot see like this because it is all in micrometer size, nanometer size. So that through that, if you see the side view will be a trapezoidal one. It will be 54.7. How to get this 54.7? Because of Miller indices. You take it is a 111, you know, we have got 110, 10, like then if you take 111 tan inverse of root 2 by 2, you will get this popular 54.7. So that is how you will take it. Then we have got, see now if I show this to any microwave person, they will not say whether it's a MEMS or BioMEMS or RF MEMS or microwave. So in the planar wave view, it will be ordinary patch antenna. Whereas in the side wave only, you can say, you can find out the distance. And of course, the size also you can say whether it is a MEMS component or whatever component is there. So this is a general rule for antenna. Antenna. How to do the design? You should not refer to the books for the design values. You take the dimensions, everything in terms of lambda. Just 1 minute, please. Okay, if a person is knowing how to do this bio-implantable antenna, the same philosophy can be extended for all the things. What all we want is how to know what is length, how to know what is the width, micro filters, MIC filters, like that. If you know the principle, you can easily know how to do the design. As an engineer, every gadget what you do, you should know how to design. Design means not a very big thing. How to know the length and the width. So I will show how to do it. Now, if you take a BioMEMS, the diagnosis is not a very big thing when comparing to therapeutics. And therapeutics also we are very good, especially if you go in India. I'm not telling India alone, many places therapeutics also being done. That is a great wonder. Suppose if you go for a telemetry, sitting in one place, curing a person in a village, that's a great thing. So that is what I am telling. If a thing can do in therapeutic side also, definitely that is to be followed. Diagnosis, it can do a big role. Diagnosis, many things can do. Therapeutics also it will play a big role. Then therapeutics, how it can be done? By the drug delivery. With reference to drug delivery, what I want to tell, suppose if you say, we keep talking BioMEMS, so many things. Can we see that, especially in India? Not only in India, world, only in few places you can see all those things. In many places it is in the research level. In some places, because there are many reasons why directly it cannot be used. Even the COVID vaccine, it took so many days for that to come to common man. There are reasons for that. So that way, what one can easily say, the drug delivery, you can see in action. Maybe if you go to, I can say a few examples. I cannot say that is the only example. If you go to Amrita Hospital, you can see the drug delivery through BioMEMS. The ordinary conventional drug delivery and BioMEMS drug delivery, there are so many important differences. I will show you. Then real-time monitoring. See, when they go for the heart implantation or heart surgery, real-time monitoring is there. Then store forward monitoring is there. So these are all the words depending upon the practical applications. So that way, in general, BioMEMS device, there are two primary aspects: sensor, actuator, and the system. So these are the places where they play a very big role. Comparatively, it all started only after 2003. I will tell you what happened in 2003. Incidentally, if you go and see the history, you will come to know about the subject also. So she is said to be goddess for the whole world for the microwave integrated circuits. They play a very big role in the design of the BioMEMS component. Her name is Bharati, but you can know much more about her in Dr. Kalam's book, Wings of the Fire. So this is one thing, bioelectromagnetics. See, normally computational electromagnetics is a brainy work. If you don't have any facility, you can, if you are very good in that computation, you can do that. The same thing if you extend to the biomedical side, wonders can be done. In fact, Nokia, what is the effect of biological effect to brain? Many things can easily be done if you can extend the electromagnetical computation facility to biomedical side. See, computational electromagnetic itself is a very big thing. You should understand a lot. The Bible for that is the book written by T. Ito. That if you extend to the biological effect of microwaves, you can predict the thing. Prediction can easily be done. For example, if you take an ENT doctor, they go for a different kind of deafness, nerve deafness, conductive deafness, many things. So that an ENT person should not waste the time in doing all those things. A paramedical person can do all those things, draw the curve, do the computation, give it to our kind of algorithm, take the output. Finally, it will give what to be done. That is what your scanning mission, imaging mission, everything is doing. The application of computational electromagnetics, biomedical side, that is the real outcome of your CT, MRI, ultrasound, SPECT, everything. So that you can even try, not in a very commercial scale, at least from the research point of view, you can start doing it. From the one what you can know what it is, all those things. So this is what that, just to emphasize that computational electromagnetics in biological side can, small example, many may be aware of finite element method. Using finite element method, burn injury can easily be told, not bluffing like what is happening, 30%, 40%, 80% like that. Pinpointedly, you can say how much is burnt. It is not for a court or anything. If you know the percentage of burn injury, you can know what is the methodology to go for the curing. And this everyone knows antenna. As far as the spacing is concerned, if the spacing is not a constraint, this is the best antenna because it uses see, this is another way of telling conventional method is the best thing everywhere you go. See, nothing like equivalent of writing myself right on the board. All these techniques I am not seeing, you are not seeing me. So whereas blackboard writing is the best methods in certain things, conventional method is the best curing the disease without any artificial interference. That's what we say, non-invasive surgeries, invasive surgery. That is the best. If not, then you go for invasive one. Why I am showing this is this kind of antenna cannot be used for biomedical purpose, especially for satellite or a small miniaturization is the mantra. For that only BioMEMS started. This is a implantable antenna, biomedical antenna. As we want to go smaller and smaller, micro, nano are the only choice. There only micro electromechanical system, RF MEMS, BioMEMS play a very big role. Before 1980s, it was the computer revolution, then communication revolution, then from 2000, healthcare revolution. And in MEMS, in all the technology, the speed with which it is developing in a very fast way, it is the BioMEMS only. BioMEMS, by the time I finish this lecture, many new things will come out. So that way, it is progressing. It is revolutionizing. There is no field in the medical where it has not invaded. Minimally invasive surgery, in certain cases, it becomes non-invasive surgery. Micro total analysis, isn't it? You take a heart. Heart is just like a hydraulic. You just find out what is the analogy of the heart with your hydraulic pump, isn't it? So that way, I do not know. Artificial intelligence is there, machine learning is there, everything is there if you take a human being. So likewise, you take a cardiovascular, diabetes, endoscopy, neurology, oncology, ophthalmology, tissue engineering. You tell a subject, there will be some involvement of BioMEMS. In fact, these are said to be the emerging BioMEMS. This was told some years before in a very popular book, first book on BioMEMS. But now so many things have come. You can yourself see many of the advancements in the state-of-the-art hospital in India itself. You don't need to go to abroad. All those things. See, day before there was a session of yoga. Everyone told, I am feeling cool, I am feeling all those things. It is electromagnetically proved. So why am I showing in this BioMEMS class? Because each one, see, it is again in terms of the lambda. Because of the different region, you've got different electromagnetic field. It's all in terms of lambda. Because of that only each color in the electromagnetic strength refers certain strength. So because of that only, so you take meditation, anything. So I want to tell you, in space application, in biomedical application, in IT and spirituality, India's tops in the country. Everywhere, what is common thing is electromagnetically, or if you call it as in biomedical, it is bioelectromagnetic. In the meditation, any type of meditation you take, definitely at one time, resonance will take place. So biomedical effect will take place. And that's what we call it as a mental telepathy. So it's a different thing. So mental telepathy, you can do in a non-invasive way. So the first day when the chief guest came, and he is the director of your biggest cancer research center in the Silchar, and the third person's teacher was India lost Dr. V. Shanta a few days before. I call her living Mother Teresa. Why I am showing here? If you happen to go to Adayar Cancer Center, you can see the latest instruments by which the initial percentage of cancer is getting cured. How it is getting cured? By the selective target. That is where this BioMEMS is very much useful. It is not to be affected in the outside the region. You should locate the place where you want to pass the microwaves. There you pass it. Heating effect, biological effect will take place. not a 90% cure cancer will get 30%, 40%. That initial stage, if you find it out, it can be cured. The LINAC, linear accelerator merged with MRI, 2-3 instruments merged. That kind of instruments you can see in such good big hospitals. So there, this BioMEMS technology directly people are now, it has not come, but related technologies being used in the form of CT, MRI. First, you should diagnose, accurately find out where it is, then pass high frequency. So who said cancer is not curable? If you can detect it early, then act immediately. It all started in 1952. From that time, Dr. Shanta was the founder person. This is the principle of cancer detection. Once you detect it, isn't it? The presence of virus makes the cantilever bend. Incidentally, I would like to ask you, people, especially the beginners, you should know the difference between a cantilever, beam, and also diaphragm. See, we have got one bookmark about the pull-in voltage. Everyone knows that pull-in voltage is the least in the cantilever. So this I am showing in a enlarged way. It will be very, very minute size. So there the virus may that is bent. This is called the selective killing. That is where it is different from other methods. You find out accurately, you find out where it is, because the other part should not get affected. That way only microwave and BioMEMS will help a lot. And this person, it is this sentence. We know what happened to Richard Feynman's. So this sentence, because of that only, we see so many ISRO, VSSC, SAC, so many things. Why am I talking ISRO, VSSC here? They have got a separate division for telemedicine technology. Telemedicine, we are number one. We are top in the country. What Aravind Eye Hospital can do, 1,000 surgery in a remote place is a wonder. America appreciated. They gave award. So this ISRO people has got a separate division for telemedicine. If you want to know much more than that, the head office is father of telemedicine is S.K. Mishra. He is in Sanjay Gandhi Postgraduate Medical Education Center. One should go there and see what is happening. You can see what is happening inside a theater by sitting outside. A very big cinema theater is there. You can see everything what is happening. You can see what is technology is doing in the biomedical side. And as he told, Indians are second to none. That was the proof of Mars mission. And another person, Dr. Ram Gopal Rao. I think some of your participants are associated with Dr. Ram Gopal Rao. And he has established unthinkable center in, you call it as INUP, it is in Bombay, then bifurcated to Bangalore also. Many of the gadgets I will show you. BIMS fabrication, usable in the biomedical side, also can be fabricated. It was he who proved that in India also you can do wonders of that. See, these are the temples. It's not only temples. I can show few mosques, few church, like Velangani Church. See, each place is known for some biomedical application. People, seismophilia, they don't, or people say mental asylum, isn't it? There are some temples assigned for that. It has got some effect. People say those people you can go there and stay for 10 days. It has got some effect. These are all biomedical, telemetrical effect. So this has got, say, if you go to this church, I don't want to name what church it is. You can see so many organs, leg, many organs. They would have made silver and put it as a praying one, because they would have prayed that defect should get cured. Once it gets cured, they tag like that. There are incidences where it happened. People that time did not call it as a biomedical. They did not call it as a BioMEMS. It is a non-invasive method of curing certain things. So that's why I showed that those temples, church, mosques. And this person, I tried him to join today. to give you a small talk of 15 minutes. Somehow I started conversing only some half an hour before, so that's why he could not join. Anyway, I will make him join with you. He is a 70 years person, solitary man. His book is the first book on BioMEMS. I wish first page to the last page you go through that. Not only that you will get enough materials, not only enough material, you will come to know what are all the gadgets, what are all the instruments needed for BioMEMS fabrication, BioMEMS measurement. I will show you all these site, and I will make Solitary Man to interact with you also later on. So he is an MD. Initially he studied MBBS in a good college, then MD completed. Then he had a collaboration with NASA. Then at one time he got an invitation from Apollo. Neil Armstrong, he sent an invitation, join in our team. So he sat with a mission director, surgeon. So as and when you move, I found Satish, I find Satish, one of the organizing person, keep talking, ask so many questions to the nephrologist. So like that, if you keep moving with many multi-speciality doctors, you can do wonders of technology in those specialty. That is how this Silchar located, NIT located Silchar, is doing a lot of work. Whether there is a facility there or not, you have to get associated. They should give you input. And from that, you can see what best you can do from the engineering point of view. So that way, he is a medical doctor. See, the back, you got so many other instruments, data analyzer, spectrum analyzer, audio oscillator, noise figure emitter, all those things. That is the real marriage of medical to engineering side. So this is the book. See, in this short time, till 1 o'clock or 1:15, you can note down certain references that will take you to this BioMEMS path. And this is the first book. There are one more book on BioMEMS I will show you. And here he has touched upon all the branches. Urology, ophthalmology, gynecology. Gynecology he didn't touch. Cardiovascular, all those things. He has touched. Not only that, he will show you. If you go to the website, you can see those instruments. Most of the instruments you can see if you go to INUB programs of IIT Bombay or IISc. And the next book, RF BIMS. And that I should feel proud that this being written, all 4 authors are Indians. The first author is not even ECE person, he's a mechanical person, Vijay Vardhan. Other person, Vinay and Jose are electronics person. And this is a reference book for the whole world for RF MEMS. There are many books on MEMS on specific topic, but this is a book on concise one. That principle, if you can understand, that you extend it with biocompatible material and design it, then you can come out with some BioMEMS device design. If you want to do the fabrication, then you should go to such centers. The second book also is a book needed. Now it has come in the Indian edition also. So this is the website. You can know many things. Especially people will be knowing many theoretical one. But if you want to see all those instruments, from which you can get all those gadgets, this is the only slide, and also I'll give you. So this is one such center where again, incidentally, you all will be happy to know if I tell who is this person. He is also, his name is also Bhattacharya. I think, in fact, in the previous book, one of the reference given is A. Bhattacharya, who gave a lecture 2 days before. So this person is another Bhattacharya. He is the person in charge of this MEMS fabrication lab. This is, in fact, the same thing only you can find out. I think many of you might have gone inside such kind of lab in INUP Bombay or in IISc Bangalore. And one such person is also giving a lecture on the last day, Dr. Anand Suresh. And this is one of the investigating station. Then they do fabrication. This is the center from California State University, Northridge. Incidentally, that dean was my student. That's how I could go anywhere. So next, these are all the instruments. These are the gadgets. You can see all the gadgets if you go to the website of INUP Bombay or INUP Bangalore. Many of them in INUP Bombay, you can see more. The miracle, marvelous, has been done initiated by Ram Gopal Rao, who is presently IIT Delhi. So this is myself. I saw only few. Initially, you should know. You should take the weight of many of the ingredients, and we say clean room. There are few clean rooms. See, seeing is believing. Whenever possible, don't go for the bookish knowledge. Go and see the reality. That is how few people are shining in India in practical things. I can very well say, but let me not point out some names. They are not shining because they all got to the reality. They have got to the place where it is being done. So now this is not in California. This is the scenario in IIT Bombay. There are some clean room. The smallest clean room is in NQIL Kochi. Cochin, in clean room, people will say 1,000 or 10,000 like that. The biggest clean room in India is in SCL Chandigarh. SCL Chandigarh, that was the dream work of Professor Atre, the eminent person who is instrumental in all these things. Professor Atre's contribution and Professor Ram Gopal Rao contribution. Because of that only we enjoy the reality. We can also be proud that India is doing all the things. So these are the gadgets. Again, this is not in abroad, this is in India. So this is the, you can see how to fabricate, how to do investigation, all those. So this is, you can see how much it is shining. In this small wafer, so many cells are there. All those cells, it is all in terms of lambda. This much length, this much. So we don't need to say low, high, more, large, small like that. We should tell only in terms of far less than lambda, far greater than lambda. We talk about micro, we talk about nano, all those things. So this is one such clean room, MEMS lab, but I will not be able to show any BioMEMS lab. It is not there. The only thing is BioMEMS lab in India, but I can show you some BioMEMS lab if you go to the website of that solitary man. Okay, these are all the few techniques. Suppose if you do not have much facility, this is another method, but the dimension should be up to millimeter you can do, but not micrometer and nanometer. So up to millimeter or centimeter, you can simply take use of scissors or iron box or fan and then cut it and paste it and then go for the gadgets. So this is the small example to show that how small it is. The whole system, this is called system on a chip. Now lung on a chip, heart on a chip. Now why lung and heart? Body on a chip has come. All those things you can see not in the books, in the laboratory. If you go to the website, some of these things you can see in India in IIT Delhi. IIT Delhi, they do not cochlear implants, implantable one. That's only one defect. If it's an implantable one, people may not know that that person is deaf. If he's not knowing he's deaf, how they will be careful? Like driving people, unless they put the learner's license, how the others will know he's a learning person. So in any case, cochlear implant, this is very easy. Instead of playing with heart, playing with ear comparatively, I'm telling. So here, one of the application where cochlear implant, BioMEMS plant will be of much use in the cochlear implant. And this is another little detailed program. This I got from NIT only. This is where your retinal processes can be done. The artificial lens can be implanted. So that again, BioMEMS can do. See, incidentally, MEMS, RF MEMS, BioMEMS, micro optical electromechanical system. That's again a different branch. What it can do in the optical. So there is no need that every time I should say BioMEMS. MEMS in biomedical, that's what many people also say. Maybe if you want to go in detail, then we can precisely say BioMEMS. So that way, MOEMS, that also will play a very big role using optical waveguide. Again, I can say, but he has not done much on the medical side, but in the optical electromechanical system, there is a person by name, again, he is from Mysore, but settled long in abroad. Oran Simon, he has played a very big role. Likewise, you know very well, COVID, then AIDS, then the next dreadful disease is the Parkinson disease. Now, this mechanical person, Vijay Vardhan, has come out with a gadget which will reduce the negative effect of Parkinson disease. In if you go to that university, you can see that. So this is what you can done for ophthalmology. In ophthalmology, also the optical MEMS play a very big role, and this is just transforming. Within all super specialty and costliest instrument will not be there in a village. Why village? Even in a big city, it may not be there. In a referral hospital, it will be there. So that referral hospital can get connected to any other village people. So there you can easily send the channel and you can do the diagnosis and what to be followed. That the super specialty doctor can tell the MBBS doctor, he or she will carry over that. In any case, ophthalmology also, see image processing. We say M-Health. By using cell phone itself, you can do all the diagnosis, especially in Silchar, that's a separate, maybe if possible, you can visit in Shillong, Negrins, Northeastern Indira Gandhi Medical Research Center. There also you can see, you cannot see BioMEMS, but you can see what the telemedicine can play a role there in some of the speciality of medical region. So this is the diathermy. Diathermy, a general word given for heating effect. There are some, you might have seen Philips infrared. When you have the headache, they pass Philips infrared rays. You will, in 5 minutes, you will not feel the effect of heat, effect of headache. The reason is you will start feeling the heat. So that same pain is there, but heat will be predominating. So that way, heating in the medical side, we call it diathermy. If we use microwave, it is called microwave diathermy, millimeter wave diathermy. So that way, this is the method of heating. You can say ablation. Ablation also a partial non-invasive surgery. So that can easily be done in the MRI. So this is what is done in the endoscopy. In the endoscopy, see, whatever the final year BTech student study, if we put that in the medical application, that will aid in the BioMEMS devices also. So like signal processing, ECG, EEG, EKG, EMG, all those physiological signals can easily be understood if a person is strong in signal processing. Likewise, image processing, instead of doing always the research on Lena's hat, you start doing the images of the human being, then wonders can be done. That is what being done by GE, Siemens, Toshiba, all those people. So your CT, MRI, all those things. And which technology will save all those CT, MRI? I will show you the technology called FSS. And this what you are seeing is the endoscopy. Previously, a topmost doctor, he might not be a specialty in any of the particular one, but by experience, he can say where is the defect. Now, what is happening? Any place, anywhere in the body, one can easily see. You can locate it. You can go for the optical method also without even cutting. You can go for a method of surgery also. That is the recent one. But there are certain things which the recent using optical you cannot do. For example, delivery, delivery, certain things, conventional method is the best and that is the only method. So there are some methods where BioMEMS can definitely can help. So this is what your BioMEMS activity in the endoscopy. Then gastroentomologists, they put an optical fiber at the end. You put the camera, and wherever it has to be seen, it can see. Then you can easily do the ablation method. So he is the person, Victor Vesselabo, right? What he told in 1967 was told by J.C. Bose also. I told epsilon r is equal to 1. There are some materials whose epsilon r mu are less than 0. We call it a negative material. That is what we say beta material. The beta material also is doing lot of valuable work in solving, or I should not say solve, in therapeutic work in the healthcare engineering. So beta material, frequency selective surface, that are also doing the therapeutic side. Diagnosis is a child's play with all this technology. This is a, see, once you finish out the model, suppose if you implant certain thing, it is not like a car bonnet. You can open it and close it. Once you implant your artificial heart, first of all, you should try not implanting the heart. But with no other go, if you implant it, if you want to monitor after that, that is what we say by the term reconfigurable. If I want to tell it in a different way, you take Kalpana Chawla, Sunita Williams landing on the universe. There, if there's anything wrong going in the antenna, the scientists cannot go there, they cannot come down. So from the down, you can slightly do little voltage and reorient it. The same thing you can do it. So that's what coming in the reconfigurable. So that will do a big work in the reconfigurable BioMEMS. He is called Monk. He has developed this work called frequency selective surface. This play a very big role in the diagnostic gadgets, CT, MRI, lot of things. In what way means it will shield all those things. Those gadgets shield human being, but which technology will shield them? Shielding in what way? Especially in BioMEMS, you know, EM by EMC. See, in the airport also you can see, even in COVID also they say, if there is anyone has got artificial heart, you don't go near the scanning instrument or something like that. So it is all by what? It is by EM by EMC. So this technology will definitely prevent anything from EMI, EMC shielding. And my scholar, incidentally, I want to tell a few scholars in this forum, your research work should be directly be applicable to the society. I'm not simply exaggerating. Dr. Kaushik Guha, there are many people, researchers, whatever work they do, it is not for the PhD degree. The work they do should reach the society. Biomedical work, even if you do small thing, it should go directly to the society. COVID diagnosis and therapy, how many lives you are saving. So that way, this boy, in 2 years, he completed 10 IEEE papers. The one what he did not apply it for medical side, but if he applies to the medical side, CT, MRI can be saved. Therapeutic saving of those gadgets will be done. But what he has done, the work what he has done, he kept in the flight aircraft. So the others could not see, we could see. That's why he was awarded 5 lakhs rupees. So here, what he has developed, that can be kept in this GE. I think this is GE, General Electric. Siemens also doing a lot of biomedical thing. Then Toshiba also doing. It is all engineering technology, medical application. So for that, one should have a thorough knowledge in physiology, anatomy, and everything. Can anyone forget Gray's anatomy? Isn't it? Still it is there. So that way, if you want to safeguard these instruments, you need this FSS technology. So these are the instruments, not instrument, gadgets. You can find out in state-of-the-art hospital. In Tamil Nadu, in India, there are so many state-of-the-art hospitals. What it is all up to the international level. So morning you get admitted, evening you can come out. So that is how medical tourism in India is flourishing like anything. Though it is not correct. Even our medical pharmacy people, if you say you are whatever you are facing, they themselves will give medicine, even though it is not advisable. So such a way, quick diagnosis and quick therapy can be done by Indian medical fraternity, and that they are able to do only because of enormous developments of engineering technology in the medical side. In that, the latest thing is BioMEMS application. But still, if you ask how much it is used in India, I can say very slowly. But externally, especially drug delivery, you can physically see in some of the hospitals. So these are the institutions where so many things are going on. When possible, you please go there. Seeing is believing. Like SSPL, AIIMS, they have AIIMS Delhi. They have a separate room for engineers, biomedical engineers of Delhi. Like a CSIO, you can see, you can physically see a lot of BioMEMS activities are going on in CSIO Chandigarh because they have a good collaboration with ACL Chandigarh and also Postgraduate Institute of Medical PGI. And also IIT Bombay, no need to tell. IIT Bombay, they give BioMET. They conduct and they give degree, MTech, PhD, all those things. Then in DRDO Double Bangalore, there you can see a lot of gadgets. You know, our soldiers have got textile antenna. So it is impregnating any antenna, smaller antenna. Impregnating is little different from implantable antenna. So, you know, see, suppose if you have the artificial heart, everyone knows artificial heart. But what is different with the BioMEMS? BioMEMS means, see, you cannot change the battery as and when you like. So battery longevity will happen only the devices sizes are very small. So the devices small will happen only the frequency goes higher and higher. So especially in BioMEMS, in microwaves we talk centimeter, millimeter. In BioMEMS we talk in micrometer. So the battery longevity will be more in the BioMEMS technology embedded artificial heart. So that way, these are the places. You can, nothing is imaginable. Everything reality. You can go visit those places, see by yourself, and then understand things. Simply cannot be imagined. No need to imagine. You can physically see it, and wherever you go, you see it. First of all, go to the very good hospital, if possible. Especially if you go to Sanjay Gandhi Postgraduate Institute of Medical Research. Especially going inside there is not advisable, but stand outside, and that's a very big theater. Anything, whatever is going inside can be seen outside. That is how they teach medical students. So that way, you have a first-hand knowledge of the things, what is happening in the hospital, and also see the biomedical master, and see first you master in MEMS. Then check how the design can be extended to RF MEMS. Then see how it can be applicable to biomedical. One minimum thing is you cannot use any material you like. Even this certain tablets, sometimes it becomes allergy. So we have got a separate biomedical, biocompatible material. There are a lot of researches going in biomaterial. In the world, in India, it is going on. Sri Chitrathirunal Biomedical Center, it has got different centers in Trivandrum. One center is for biomaterial. They take Indian goat heart, that size and human size heart are same. So that they do research. There are some biomaterial division. They do deep research on the biomaterial. So all those things, whenever possible, you see. Then coming out with applicable what will be easier and quicker. This is what I showed in the Mark Madhu book. You should be having good knowledge of epsilon r of different material. It is not always epsilon r. you should afterwards you should change it to epsilon effective, because with reference to locality it may change, it may not be constant. That's why we say anisotropic. And between organs of man and woman also, that's a very big difference. So all those things you should see in a very layman's way. I can say of all the material, if you put the chicken inside a microwave oven, it will get heated immediately because of the epsilon naught. Each part of the chicken has got a different time to get heated. Similarly, you take a human being and see what is the effect. So don't try directly. You do using computational electromagnetics, get the values, then go for actual thing. Now rabbits, monkeys are being used. Now, if you want to use it directly to a human being, you should know risk-free method. Then only you go for testing at the final stage. So these are the other books. For drug delivery alone, that's a book. There are so many books that have come. And this is for microscale, especially the needles. Our technicians, if they want to take the blood, a very good technician is there. With a single prick, they will remove it. They will take any amount of blood. But the amount of blood they take is more. Whereas if you go for micro needle, it's a tip. A small spot is sufficient to go for a microbiological and pathological test. So that's a separate version on micro needle. Incidentally, one of my scholars did PhD work on micro needle. He had a lot of difficulty where to fabricate, where to test, all those things. But you can read the micro needle and how it is different from ordinary conventional syringe. So these are the theoretical method designing and going for the project of devices. So again, it is the micro total analysis. So you've got so many. So whatever is happening, see, even MEMS, whatever civil people keep telling, that is the smaller scale. Civil students, they are always keep talking structure. Here also structure. There also they talk beam, then diaphragm, cantilever. Here also same thing. Only thing is they talk in the meter, whereas here we talk in micrometer. Then mostly they keep telling about Young's modulus. Young's modulus is one of the important factors to go for the derivation of pull-in voltage. So that way, filters, valves, pumps, needles, everything we know. But that you put it in the human, and it will be in a very, very, very small size. So that way, the principle of working is same as the conventional one, and size only is different. And risk factor, you cannot directly test it. Even that testing with the animals also nowadays you need a lot of permission, many things. So this is another version of the thing, and this is the structure. This I am able to show in a bigger scale, but actual scale will be in a smaller way. And this is the small device used mainly in the drug delivery actuators. Actuators only three types. The diaphragm in one case, diaphragm size, other is anti-lever. So you can easily find out how much voltage required. See, even inside the body, 3-4 frequencies are generated. The cell membrane generates 34.5, 46.5, 15. So that kind of gigahertz it generates. Likewise, it generates voltage, not a high voltage. So ECG, EEG, EEG will be very less, alpha, beta, gamma, and all. Whereas ECG, few millivolt. So by that, only people find out, do the diagnosis. But whatever is happening, this question is to all the participants. Whatever BioMEMS, whatever it comes, no one can replace stethoscope and thermometer. Thermometer and stethoscope, you can also call it as a sensor only, but nobody calls it a sensor. Definitely, it belongs to the family of biosensor. That's what I told. Whatever things come, certain things conventional is the best. Only when you cannot go, see now I cannot come and see you, you cannot see me. That's why we go for this unwanted online programming. So similarly, you cannot see a doctor. See, even yesterday when he came, nephrology came, he wanted a stethoscope. It is needed, but for that stethoscope, thermometer, whatever digital has come, that will not have the same effect as the a real stethoscope and the pumping manual BP apparatus. Yesterday, what he told about you should check your BP for BP. BP makes 50% reason diabetes. So stethoscope and that. So previously scan machine was not there. Even without scanning, the experienced people did everything. Previously computers were not there. People were doing. Now everything how come we are complicating. In any case, in certain things that will ease our problem. So this is your lab on chip, then implanted devices. Why I'm showing is you see the cross section of any of the device, it will be same as microstrip and coplanar waveguide. In fact, those are all used in the centimeter by the engineers, whereas medical people, they should use it in micrometer size. So people, very young boy, an Indian boy, he only modified the existing microstrip and CPW suitable for biomedical application. You take any BioMEMS, you take a MEMS component, RF MEMS component, if you take a cross section, it will be either will be looking like microstrip or will be looking like a coplanar waveguide. So what I want to tell is some of the theory can easily be understood or easily be designed more by millimeter engineering people. So I think I will take another 5 minutes because people should not have a curiosity when I'm going to stop. So this is your FSS structure. So you know 11101 like that. So because of that one day in BioMEMS structure, you will get tan inverse of root 2 by 2. You get that magical figure 54.5. In Delhi or in India, there's a separate hospital exclusively for spinal injury, isn't it? You know very well how much cause it will do. There is an injury in the spinal cord. So there is an exclusive hospital in Delhi. You can see not only this kind of wheelchair, in the evening you will be surprised. You will shed tears. Some of the patients go to basketball ground sitting on a wheelchair. You can see players sitting on a wheelchair playing. They forget they are affected by the spinal injury. So because if you have the willpower, there is a way. You should never get depressed. So why I am showing spinal cord injury here? In a spinal cord injury, BioMEMS can do wonders. Isn't it? You take a stem cell with the help of stem cell, the damaged spinal cord can be regrown, and also use it with the electrical stimulation. Just like a diathermy, the common word for electrical stimulation is long back it is being told. It is called plethysmography. So how do you do it? If the low frequency or high frequency, by that it earns some other name. So in any case, when chance comes, very recently I had a chance, so I know what is happening. Everything, whatever possible, every one of you, please see. Hello. One more method is you should become a member in all those societies. Then seeing is easy, isn't it? First thing, what many of you, if you have not become, you should become a member of IEEE. Head office is an IIC. If you become IEEE, getting insight to all these MEMS manufacturing place in India is very easy. IEEE. Likewise, Telemedicine Society of India, then EBMS in IEEE. So if you become a member, fellow, senior member, the approachability of such places. Otherwise, you may not even be allowed inside the gate. So you please become member of all those things, especially these MEMS, BioMEMS interested people. Become member in IEEE. you come to know many things what is happening in India. Not only knowing, you can physically see all the places in India. Otherwise, getting inside is not possible. How I can say Chitra Tirunal, who will allow you inside? Well, they should know you deserve to go inside. So that way, that is two things I want to suggest. I don't want to advise. You try to become professional society member, especially in this subject, IEEE, EMBS. In fact, there is another society, IEEE microelectronics also. Then TSI also. Then second thing, let your research paper be useful to the society, not aiming to get a publication of the paper alone. Then the interdisciplinary, not only the engineering interdisciplinary, you have a good interaction with medical people. In this meeting, I could easily find out both should be equally interested. I find the nephrologist is very much interested to interact with this NIT Silchar people. Likewise, I found one of the scholars, Satish, very much interested. Then only you can know. The book reading alone will not get all the informations. So in another 2-3 minutes, I will stop. In addition to the known method of fabrication, there's a own separate book also written by the same set of people, microstereolithography. That is slightly better than other conventional method of bulk surface and liga. And polymers, PMMA. So polymers has got a separate entity to be used in the BioMEMS. This is the reason why we use microwave technology. We say for medical people, I simply will call high frequency in the reason of gigahertz, terahertz. This is a small. I don't want to advertise. From the first principle and writing the programming by your own code, either MATLAB or Python, not using the commercial software. So you're having one IntelliSuite, he has got a special interest. The person who is coming day after tomorrow, he has got a special interest in designing BioMEMS device. So with this, see if you see this cross section, this are all the etching process of the BioMEMS component, but it looks like a slot line. It looks like a not looking like, it resembles a microstrip CPW. These are all the IC, MIC. The same thing, you go for a higher frequency, use it for a BioMEMS component. So the previous one is microstrip. This is a CPW. These are the cross section for a BioMEMS component. This is the 1 or 2 slides last. Using artificial neural network. Using artificial neural network, you can do wonders. This is a small example. Get the knowledge, expert system, get the knowledge, use your expert knowledge, deep learning or machine learning, and then see if you want to go to a cardiologist. Get all the 64 possibilities. Put it in a artificial, make it as an expert system. Use the computer programming and any measurements got from the ECG, PQRST, if it is fed, it itself will tell you that patient belongs to what category. So you need one expert person to tell all the heuristic, then you should program it. So that way artificial neural network can be used for any of the specialty. I think with this, I would like to close my general overview talk. Thanks a lot for the patient hearing.
Thank you, sir. It is obviously always a pride moment and very happy moment to listen to you, because those who listen to you and your talk, so they might know well. So you have given a very pertinent and very highly interesting overview of all the BioMEMS domain, and you have shown that how the with the limited facilities, how people can start. People can start the BioMEMS work, the research in BioMEMS, and it is a good blend of microwave and also and with the MEMS, because you are a very quite well-known and experienced person.
Hello. Very happy to know that one participant from Gurgaon, he has given the address of that spinal injury hospital. It is exactly in Vasant Kunj.
Yes, sir.
Institute of Spinal Injury. I hope you have seen
Yes,
that
sir.
hospital.
Yeah, yeah.
If you become member, they will take you inside the theater also.
Yeah. Yeah. They have a great facility here.
So what I told is correct or not? You tell me.
Yeah, yeah, he's right. There are a lot of facilities as well as if you see the Subsurgeon Hospital, they have started a sports injury center during Commonwealth Games. So that also I have given the link. You can find a lot of facilities there. Hello.
Okay, thank you.
Yeah, thank you, sir.
Yeah. So So now we can take few questions because so we are at 1:15 o'clock. So so that's why we will not ask all the questions to our honorable speaker. We can take some one or two questions. So because Sir has given a wonderful presentation, and those who want to connect with Professor S. Raghavan, then they are always welcome to connect because Sir is very also very cooperative and very happy to assist with all of us with his wide knowledge and experience. So now we can take just only one or two questions. If you have any specific questions from participants, then you can unmute and just ask the speaker. So any questions from anyone? So please you unmute yourself and can ask.
Hello.
Yes, please.
Yeah.
Yeah, good afternoon, sir. I'm Dr.
Yes.
Anita. I'm glad that I got a chance once again to hear your lecture. It was beautiful presentation. Right, and I have one question, sir.
Ye-
You discussed about metamaterial antennas, that means metamaterials. So that
Yes.
can be used for biomedical applications. So my question is, particularly, can we use this metamaterial for cancer?
No, this metamaterial that can be used for diagnosis purpose. It can no, but for direct for therapy, it cannot be used. But still, I have not come across metamaterial in cancer, but it can be used very well in the medical side. If you just Google metamaterial, but in fact, there's a book coming
Okay, sir.
by Shivanarayanan. In that, there's one chapter I have written what
Oh.
metamaterial can do in medicine.
Okay. That's great, sir. Thank you so much, sir.
Yes. So any other questions? Only one or two, maybe maximum two. We can take.
Thank you so much, sir, for your enlightening talk on make us aware about the facilities across India. So, can I know that what are the work you are doing now at NIT Trichy?
Okay, with reference to
Cross
BioMEMS,
area, yeah.
I am not doing much work. Only one scholar of myself did work in the micro needles.
Okay.
That was the real work, fabricated work and other thing. Otherwise, I have a hobby in visiting hospitals.
Yeah.
My subject is microwaves, but this biomedical side is visiting hospital and seeing what is happening.
Yeah.
And few
Thank you so much.
mechanical problem I solved.
Thank you for your enlightening talk for the young generation. Thank you.
Yes, sir. Yes, sir. Sir is a microwave expert, but basically, I think so. We can, if we can combine the microwave with MEMS, so there is a wonderful application of these two, because these two can be suited with each other, and there are a lot of research and good innovative work is going on the blend of microwave versus MEMS, RF and microwave MEMS also. So with this, I think no more questions. So now so Preeti, you please
Sir,
conclude. Yes,
I have a
think.
question
Please,
if that's
Oh,
some time.
Acha, you have a question. Please ask. Yeah. Last question.
Professor Raghavan, sir, it's a delight to listen to you, sir. Sir, you
Yes,
were
please.
mentioning about the skin burns, like it's 30%, 80%, flukely saying like that. Other than that, if a device could actually measure how much of burnt percentage it is. So could you throw a little light on how you were expecting that device to actually locate the burn percentage?
Okay, see, I will send you the paper. I know you are a,
Definitely,
even though
sir.
you are a scholar, I understand you are a professional MC also. I will send that paper.
Yeah.
So it's a combination of a finite element method.
Okay, sir.
You should make it into smaller structure.
Yes, sir.
Maybe the golden rule is to make it 1,000. You make it a small structure, then do the analysis. That way,
Okay.
more than the medical, more than the societal point of view, in the hospital, people say different, 90% cannot be good. But
Mm.
for a curing, you should know what percentage. So that way,
Yes.
that helps. How it
Yes.
is done, I will send you the paper, FEM.
Thank you, sir. Sir, with this input of like a 1,000 that you have given, may I ask one more question? So this golden rule of 1,000, is it fit for the micro electromechanical systems as in common for the FEM?
Okay, see the real thing. If you want to have a curve, even with the 3 samples, you can do it. Then it will be a discrete curve. It won't be a smooth curve. So maximum is better. But if I say 10,000, it will take longer time. CPU time it will take more. If it is
Many
very
cycles.
less, you may not even get a curve. It will be simply a discrete kind of curve. So that way, I told. But exactly there is no hard and fast rule. It is not the theoretically 1,000 is not the thing. So that you can know from the uh, different technology of the what do you call training. Isn't it? When you go for training the neural network, there's sometimes 100, sometimes 500. So that you can find out which is the member for the respective system, back propagation, Levenberg. For each, you should know which is the fittest number. But in general, higher will be the best result. But you don't need to waste time. But lower will not be a smooth curve. So in between, you will take a number. So that many times.
Yes, sir. May I take this one more question, please, sir? So you were talking about the neural networks.
Yes, yes.
Yes, sir. So I'm working on Parkinson's disease, sir. So it's all related to the neurodegenerative kind of thing where people would not be able to communicate, the brain would not be able to communicate to the system properly. So could a neural network training actually help these people in any way, large or small scale?
Okay, see, from neural network, theoretically it will be possible to know what is the best method of training and other things. Practically, if you want, because you should make this person face a lesser effect, then so you should take the help of stimulation, other things also. But as it is, the effect of neural network for paper, for research paper, you will get it easily by forgetting the other realness of Parkinson's disease. Take use of the ANN method and then finish it out. If you want to see you should make use of other physiological conditions, practical conditions, and little more who has achieved this. Only thing is it is not commercially. The author of that book only, RF MEMS author, Vijay Varadan. You go to his website and see how he has come out of this Parkinson's disease in the reality. Still, it is there in the lab only. It has not come to the commercial side, but since you have asked that question, you can go to the website and see Parkinson's disease, how it is cured. It is not cured, how it is reduced. The effect due to that is reduced by his method.
Thank you, sir. Thank you. So with your permission, Guha sir, may I conclude the session?
Yeah. So I think so as per the wish of the Professor Raghavan, so we want to now at the closing ceremony, at the closing of the session. So now we want to felicitate again our honorable speaker, Professor S. Raghavan sir. So because we could not felicitate at the beginning, so I will not missed the chance to again felicitate. So thank you, sir, for coming and joining and accepting our invitation. We want to felicitate you with our traditional Assamese gamucha and the flower bouquet. Please accept our felicitation. Thank you very much, sir, for your talk. Thank you.
Thanks a lot. In my life, I have not got this kind of felicitation.
No.
Really, everyone enjoyed.
Thank
All
you.
the participant speakers, they enjoyed the way in which you conducted the inaugural function. It is not I told, the director of your cancer research center also told. Wonderful way of felicitation.
Thank
Very
you.
nice.
Thank you, sir. Thank you. Now, Preeti, please conclude.
Thank you very much, sir. It's indeed more than a pleasure to me to actually listen to your talk personally. And I thank you for placing before us a plethora of ingredients related to BioMEMS, MEMS, RF, MEMS, and MOEMS. And so thank you for introducing us to many personalities like Dr. V. Shanta, Dr. Steven Saliter, and introducing us many real-time laboratories like the INUP in IIT Bombay, IISc Bangalore, FCL Chandigarh, and Sanjay Gandhi Postgraduate Medical Institute. And also thank you for pointing us to many books and authors like MD Sebastian, OP Gandhi, Tieto, Dr. Steven Seliter. Sir, thank you so much for enlightening us in the real time about the suffering of the patients related to spinal cord diseases. And thank you for throwing light that our research has to relate to the society directly. It's a reminder again for us, and maybe really do a research that is not just limited to papers, but would really get related to the society. Yes, sir. Seeing is believing. We have seen you. We now believe why it is said better than 1,000 days of diligent study is one day with a great teacher. We are indeed fortunate to listen to you, sir. It's so nice to see you, sir.
Okay.
Thank you.
With the permission of Dr. Guha, one minute I will take.
Yes, sir. Yes,
I
sir.
think
Please,
this is a special instruction from the coordinator, Dr. Guha, to all the MCs. What I observed is, of all the participants, I think that the MC listens from the beginning till the end, because this is again a different thing. MC not only connects from the beginning till the last, that person is telling what is happening in that lecture. That's what I observed in Mr. I think he will become doctors very soon. Dr. Satish, Dr. Preeti, other two persons also, while they are coordinating, they are telling what happened in that lecture. So by that, whether it's a good compulsion or bad compulsion, they are compelled to hear every word they tell in the lecture. That is again a unique one in my 67 years of life. So once again, I congratulate the organizers. And as I told in the inaugural function, there are many participants who are stalwarts, that people, including the nephrologist, wonderful person. And Dr. Guha, the youngest BioMEMS, the youngest MEMS person I know in India. Please, you got fortunately, you're having a very good director. So please establish the, before these people, these MCs people, I think 4 people are with you. You finish off establishing the Center of Advanced Study. Let it be the first institute to establish BioMEMS. Before KLE does, you please finish it off.
Sure.
Because for
Thank
you,
you.
government
Yes,
will
sir.
help you a lot.
Yes, sir. Yes, sir. Obviously, with your guidance and support and cooperation, obviously, sir, we'll do it. So I need your guidance always, all the time. So obviously, sir, I will talk to you regarding that. So now, thank you very much again for you, sir, for your time, because I know how much you are busy and what are the other activities you are involved, not in just in education and the research and this. You have a well the spanned the research domain, then the microwaves, the millimeter waves, the RF MEMS and everything. So it was a really privilege, and again we will invite you in some other occasion in future, hopefully in the off-campus mode and physically. again you will come to my campus and visit and interact with me, with our department colleagues and my scholars. So thank you very much. Thank you for your time, sir. Thank you. Thank you, sir.
Okay, thank you.
So the participants, we will meet for our next session at 3 o'clock. So kindly come back at 2:50 so that we can start our session 3 p.m. Obviously, this session after the lunch will also be very, very interesting one. If you can see the title, the artificial pancreas system, the role of computation and the control. The Dr. Rajiv Dey is very accomplished researcher in the electrical engineering domain in our institute, NIT Silchar. He is doing a wonderful work on computation and control. So I think it will be a nice experience for all of you to listen to him. So thank you for joining the 2 sessions in the morning. And I hope with the same zeal, you will come back and join the last session of today at 3 o'clock. We'll start. Thank you very much. Thanks. Yes, the next one at 3 p.m. Okay. So there is a time. Please come back at 2:50 so that we can start at 3 o'clock. Thank you.
Fellow members, please join me in giving the most cordial welcome to respected Dr. Rajiv Dey. Sir, a warm
Yes,
welcome from Department of ECE and NIT Silchar. We are glad to have you with us this afternoon. I humbly request you to accept our hearty felicitation.
Yeah. So, Dr. Dey.
Thank you.
So, yeah. We are trying to felicitate you virtually because we could not host the program physical mode.
I know it.
Due to the situation, because though actually you are in the station in NIT Silchar, but still actually we are unable to felicitate you physically.
Yes,
so
yes.
kindly accept
Yeah.
our
Sure,
virtual
sure.
felicitation.
Thank you. Thank you.
With the traditional, our Assamese gamosa and the flower bouquet. So thank you, Dr. Dey, for accepting my invitation and giving the very valuable time to offer a good talk on these workshops. So,
Thank you, sir.
Sir, thank you for accepting our felicitation. We are so glad to have you here, sir. I take this opportunity to introduce to you Dr. Rajiv Dey. Dr. Rajiv Dey is Assistant Professor in the Department of Electrical Engineering, National Institute of Technology, Silchar, Assam, India. He obtained his BTech in Electrical Engineering from NIRIST, MTech in Control Engineering from IIT Kharagpur, and PhD in Control Engineering from Jadavpur University. He did his postdoctorate from the Department of Atomic Control, Center of Research and Advanced Studies, Mexico City during 2015 to 16 under the grant from TWAS, Italy, the World Academy of Science, Italy. Dr. Dey has more than 20 years of teaching and around 12 years of research experience in the area of control and applied mathematics. He has guided 4 PhD students so far. He has obtained sponsored projects from AICTE, Spark project from MHRD, Project Matrix from DST, and 4 GIAN projects from MHRD. During 2018 to 20, he was visiting faculty under ERDS MUS Plus program of European Union to UAV Romania. In August, he has been appointed as a visiting faculty by UAV Romania to undertake collaborative research and academic activities with the university. He is a senior member of IEEE since 2013, FIE since 2021, member IFAC Technical Committee on Computational Intelligence in Control since 2020. Executive member of ACDOS, Advanced Control and Dynamic Optimization Society since 2014. He has volunteered as IEEE Kolkata Section Executive Committee member from 2014 to 17, branch counselor of IEEE STB PES chapter of NIT Silchar 2014 to 20. Dr. Dey has published 5 books and research monograph with Springer. He has published 30 research articles in SEIU journals, around 16 research articles in reputed conferences, 5 book chapters, and published 1 Indian patent. His research interests are time delay control, control applications in biomedical engineering, machine learning, and data science in control engineering, and physiological modeling and dynamics. So we are glad to have you here this afternoon. Thank you for accepting our invitation to deliver this talk. The stage is yours. So please.
Thank you very much for the invitation and the introduction. So can I share the video? Can I share my screen? Please tell me if you can see it or not.
Yes, yes, yes.
Visible, na?
Yes, visible.
Okay. So without wasting more time, let me start with the topic. So as the FDP is on BioMEMS and devices, so I'm little off track from the theme of the workshop. But anyway, I will not go into the mathematics of control, but I will just give you a bigger picture of what is this research and where these MEMS devices actually find its way in making up the technology. Though I do not work on the hardware technology, I work on the control algorithms part of this project. So maybe by the end of this lecture, those who work in BioMEMS can identify an area of research where there can be huge contribution from this society towards making an artificial pancreas system a reality in India, which is still far, far away from reality for the developing nation. So let us start. Good evening and good afternoon to all of you. What happened? Slide transition is not taking place. Okay. So this will be our outline very briefly. Understanding the diabetes disease, which most of you know, but some nitty-gritty we'll see. And I will introduce what is artificial pancreas system. Is it a bio artificial pancreas or non-bio artificial pancreas? You will understand. And then some mathematical modeling that is very important for control people and who develops algorithms for the devices to work. So we need to understand the mathematics behind this glucose insulin regulatory system of the body. And then some of the control problems that needs to be solved, and some interesting video across the world how people have implemented the technology. These are some acknowledgement to the contributors with whom I have worked in the past and still working. Anirudh is was PhD student of NIT Silchar, currently working on certain technologies in IIT Delhi related to biomedical. Pancreas, most of us we know, a major organ of our body. So biologically, it's our focus. Pancreas is both endocrine and exocrine gland. But when we talk about diabetes, pancreas main functionality as endocrine gland is important for us to understand. So here we see that it's an endocrine gland. because we talk about diabetes means we talk about glucose as an hormone in a body, and that has to be secreted directly to the blood plasma or in the blood directly. So we call it as an endocrine gland. So we focus on the endocrine functionality of the pancreas of the system of our body. So we know that as an endocrine gland, it will regulate the blood sugar or glucose of our body by secreting certain hormones, insulin, glucagon, pancreatic polypeptide, and others. And pancreas do participate in digestive system of the body. That is an exocrine functionality. It secretes enzyme called pancreatic juice and helps in breaking down our complex food into the glucose or the energy in the body. So this is how pancreas looks like. Most of us have not seen, of course, but yes, it looks like this. And now another preliminary that we must know in biomedical engineering is the terminology called homeostasis. Homeostasis for an engineer means that, or for any people in science, it means in a very simplistic word that a tendency of any system to come back to its equilibrium. And our body as a system also have a tendency always to come back to its equilibrium. So this particular term in biomedical engineering is what is known as homeostasis. Okay. So we know that our body has got so many parameters, and that parameters are always under control. When under control and at a particular specified level, then we know that our body, we say we are healthy because all the parameters of the body are in equilibrium or are stable, right? But if there is any deviation from equilibrium condition, and by its own mechanism of our body, it does not comes back, then it becomes a diseased body. It means homeostasis for certain parameter is not working fine. So that is the understanding of homeostasis as an engineer. Definition is quite easy to understand. So there are many body parameters that needs to be controlled, and there are n number of diseases connecting so many parameters of our body, which clinicians and so many specialists, okay, are actually giving us so many tests to find out the parameters of our body, and then trying to find what disease do we have. So in our today's lecture, our main focus is to understand diabetes. So when it is we are talking about diabetes, the body parameters are usually blood sugar. So the level of the blood sugar, if it is normal in a body because of the normal metabolism of our body, then we are non-diabetic patient. But if we are diabetic patient, or if someone is diabetic patient and we know the level of the blood sugar is not normal, right? Maybe for many different reasons, the patient cannot have a normal blood sugar. So it means homeostasis is broken down. It is violated. Condition of homeostasis is violated with regards to the blood sugar in case of a diabetic patient. So let us try to understand how the body actually maintains homeostasis in terms of a blood sugar level. Okay, so you see in this picture, when we take some food, which I have written as meal. So as soon as we take meal or food, the blood sugar level in our body rises. Okay, so as soon as the blood sugar level rises, there are certain detectors or sensors, biological, natural sensors available in our body. And they are called, and they are present in the pancreas actually. Islet, we call it islet of Langerhans. In islet of Langerhans, there are two sensors, biological natural sensors are available, which are called alpha and beta cells. So these, when the blood sugar level in our body rises, this alpha cell of our body stops secreting the glucagon. So the release of glucose in the body is automatically stopped because alpha cell senses it and they stop secreting the glucagon hormone. And the beta cell in turn starts secreting the insulin into the blood. Okay, and these functions are done by pancreas actually. So one is being stopped because the body understands we have taken a lot of food, so glucose is coming from the food. So the body by its own mechanism will not secrete any more glucose into the blood plasma, whereas this extra glucose that is rising in our body has to be utilized, and to utilize there must be a reaction with insulin, and when insulin reacts with the glucose in our body, then the glucose is being utilized or cleared from our blood plasma. So this is what is the mechanism. So what happens? Your alpha cell stops secreting glucose hormone, beta cell starts secreting insulin. In turn, what happens? The liver, the greatest endocrine and exocrine organ again of our body, liver will stop breaking glucagon, and the supply of glucose in the blood cell will reduce, will not be there. And most of our cells or tissue present in the body will increase the uptake of the glucose due to rise in insulin. Means there will be a chemical reaction between insulin and the glucose, so that glucose will be utilized from the and if the glucose is utilized because of the reaction of an insulin, then automatically the level of glucose in the blood will start reducing. So because of this action, you see blood sugar level will get stabilized after some time and we will reach to a normal blood sugar. So it means there are some sensors connected, biological sensors connected in a body, which gives this automatic control of sugar in our body. This is one scenario. And in the orange color or in the yellow color, you can see I have written, suppose we do not eat meal for a long time, or we do excessive stress or exercise, then what happens? Then because of excessive exercise, physical exercise, skipping the meal at a regular interval, the blood sugar level of our body falls. So when it falls down, the sensors again detect this rate of decrease in the glucose. So alpha cell now starts compensating for the decrease in the glucose by secreting glucagon hormone into the blood cells, and the beta cells of the pancreas now stop secreting the insulin, because if insulin is secreted when sugar is coming down, if insulin is still secreted, then the glucose will go down below a certain level and it will become a catastrophe. So the body beta cell signaling will be there, and beta cell sensor will signal the mechanism in such a way that insulin hormone will be not secreted at that point of time. So it is just the reverse action of what has happened above. And because of that, what happens? The liver now, which has stored some insulin from the above step, the liver which has stored certain glucose from this above step, now releases the glucose into the blood or blood plasma, right? So as the liver will release the stored glucose into the blood plasma, so the level of the glucose will rise in your blood, in our blood plasma, and the blood sugar level will again be stabilized and will reach back to the normal blood sugar. So I have given two situations. When we take food, blood sugar rises, signaling mechanism between alpha cell and beta cell is such that sugar is stabilized. Second situation, when we don't take food and do too much of physical exercise, then also our body is capable to stabilize the blood sugar by doing the signaling in different way, and the reverse way, and whatever stored glucose is there in our organ like livers, then they supply that stored glucagon hormone into the blood, and again it is stabilized. So now you can understand that why people get diabetes normally. People do say a lot of tension, not taking food properly. So right from the age of 20, when we are getting our BTech degree, and after that taking lot of pressure of work, tensions for next 20 years, by the age 40, 45, what happens? Lot of skipping of meals, lot of tensions, lot of physical exercise, no regularity in food habits. So continuously the body gives you a service of maintaining blood sugar for maybe up to 40 years. After that, there is degradation in the biosensors present in our body, and they stop working properly. And then we find that if we take a meal, blood sugar always remains at a higher level and does not get stabilized. If we don't take food properly in a good proper time, we find that our blood sugar level goes too low, liver is not working properly. That time we refer to diabeticians and people say that we get a diabetes, right? So diabetes is a disease at which you will have, or we will have, the level of blood sugar not stabilizing at a normal level. So now we need to understand why it happens, how it happens, and how it can be controlled. So what happens if our pancreas does not work properly? So this signaling between alpha and beta cells does not take place properly, because of which there is an open loop condition in our entire control system of glucose and insulin regulatory mechanism of our body. And as a control engineer, we call it, there is an open loop condition. So there is improper signaling between alpha and beta cells, and body cannot respond in a closed loop system. And thus, if you take a meal, and as the beta cell or insulin is not secreted properly, so the mechanism between glucose and insulin to reach to homeostasis or at a particular level cannot be done automatically by your body. So that's what is an open loop condition, and because of that, your blood glucose level will always remain elevated. Your level will be always higher than the normal level. And if we skip meals, then what happens? Again, there will be an open loop condition, as your beta cell is not working properly. The signaling between alpha cell and beta cell sensors does not work properly. And you know, to make any system automatic, maybe biological or engineering, the sensor must work properly. If sensor fails, the signal communication fails, means the variables of the system cannot be controlled properly. So because of this open loop condition, all this work that I have highlighted has to be done by liver, pancreas, and the reaction between glucose and insulin will not take properly, and you will find that the level of the blood glucose, if it is going down, will go very sharply if you skip meal or if too much of exercise a diabetic patient does. So you will find that a diabetic patient will not do too much of physical exercise beyond certain limit, because they know they cannot utilize the glucose properly in their body. And if there is too much of fall in the glucose level, you might have seen diabetic patients shivering or maybe losing consciousness. That is another very dangerous effect of a diabetes disease. So when the pancreas is impaired, basically your biosensors are not working properly, causing an open loop condition, and thus the level of blood sugar is not maintained properly. Okay, am I audible? Actually, I cannot see those screens. Am I audible?
Yes,
Sir.
absolutely fine.
Okay, okay, thank you. So now let us define what is a diabetes disease, or in biomedical term, it is called diabetes mellitus. So it is simply called in loose term a diabetes disease. It's a chronic condition when blood glucose level of human body always remains at an elevated position. And the main reason is due to no secretion of insulin from the pancreas, or maybe due to insulin insensitivity onto the blood glucose. Okay, so this is what is actually in biomedical term we call it as a diabetes mellitus. And diabetes are basically of two types. It is very important to understand that artificial pancreas is for type 2 or type 1 diabetes patients. So type 1 diabetes means the body do not secrete, or in pancreas do not secrete insulin in the body. It is a very dangerous situation. Means pancreas is completely non-functional. Beta cell is completely damaged. So if insulin is not secreted, so we can understand a complete open loop condition in the biological system, and thus there is no question that glucose can be automatically brought back to the normal position. So it is written that glucose cannot be removed from the blood. Means whatever food you take, every time glucose level will keep on increasing and it will never come down because insulin hormone is not there in the body. So such type of diabetes is called insulin dependent diabetes. You might have seen many patients who are not type 1 but extreme case of type 2 diabetic patient who take injections in their stomach, lower abdomen sort of position. So they are insulin dependent diabetic patients, and they are extreme severe patients actually, type 2. But type 1 diabetes are actually predominant in children's, juvenile. This type 1 diabetes is also called juvenile diabetes. Age group from age 1 or 0 to 13 years. It means from the mother's womb, they actually get this diabetes. So this is a very pathetic condition. Actually, if someone is born as a normal healthy baby, does not have a diabetes at that point of time, will never be a diabetic type 1 patient. But type 1 patient, normally this pancreas does not work even at a very infancy rather. Okay, normally at the birth or even immediately after the birth, actually this diabetes crops up. So and the patients who get diabetes at the age of 30, 40, they are not type 1, they are type 2 diabetic patient, and they are called insulin independent diabetes. They do not require insulin for maintaining the blood glucose, but they have some malfunctions in their body metabolism, because of which insulin is secreted in the body, but insulin reactivity with the glucose actually decreases. Thereby, level of the blood glucose always remain elevated. And there are anti-diabetic drugs by which we can control the sensitivity of the insulin, reaction sensitivity of the insulin with that of the glucose. So this is not a very severe disease. Type 2 diabetes is basically treatable with the help of diabetes management, and this is not of much concern, but yes, we need to monitor and manage the disease very well in case of type 2 diabetes. So let me clear here, artificial pancreas is not meant for type 2 diabetic patient. It is meant for a situation where pancreas is completely impaired and insulin is not secreted in the body. That is artificial pancreas for type 1 diabetic patient, which is around 7 to 8% diabetic population of the world are type 1, and around 93% of diabetic population of the world are type 2. So these are some of the WHO report on diabetes. I will not read out, you can see, I will just give you some time to read. And the most important thing is when I give lecture, I just want to spread one thing. 14th November is a World Diabetes Disease, and let us create the awareness about the diabetes in the rural India, where most of the people do not know that the cause of death in many cases, but it seems that diabetes is a killer disease with long-term implications and organ damages. Many consequences are there from a diabetes. So diabetes is now declared as an epidemic, non-communicable disease, an epidemic from WHO. So it is a matter of concern for research community like us and medical fraternity to work towards better diabetes management and technology intervention. So complication from diabetes, most of you know, long-term complications. There is a damage in the eye, neuro, nephro, related to kidney, cardiac arrest, leg amputation, gangrene. So these are the organs which normally get affected if the diabetes patient has the level of glucose, if it always remains elevated for longer period of time, and if we do not bring our glucose level at an optimum point or at a normal level, right, then slowly and slowly the organs get affected. So the life expectancy of a human being suffering with diabetes decreases considerably. So that is why WHO is concerned about it. And short-term complication, diabetic coma, series, and cardiac arrest. These are very short-term diabetic consequences. There are few terminologies which are very important to understand. One is called hyperglycemia and one is hypoglycemia. Hyperglycemia means if the level of glucose in our blood always remain above normal, then it is hyperglycemia. If the level of glucose in our body comes below the normal level, then it is hypoglycemic. Okay, so hypoglycemic condition in a body is very, very dangerous, because if the level of blood glucose comes below a normal level of blood glucose, then within seconds we may go to coma or cardiac arrest, or it can be even death. So hypoglycemic condition, to avoid hypoglycemic condition, the technology, people are working towards technology to develop a technology, automation, biological automation for type 1 diabetic patients where hypoglycemia should be avoided, right? So you might have seen diabetic patient keeping chocolates in their pocket. It is because they want to avoid hypoglycemia, because if the blood glucose falls, they become uneasy, they get nausea, they are not stable, they cannot walk, they may faint, and once they faint, they may have a coma, or if they have high pressure, then there may be a seizure completely. So immediately they take some chocolates to rise the level of blood glucose. That is the implication. And hyperglycemia does not have such consequence, but if hyperglycemia persists for 10 years, 15 years, always your glucose is above certain normal level, then slowly and slowly liver will be damaged, kidney will be damaged, long-term implications will come. So this is very important to understand, because in my later part, I may be using the terminology hyper and hypoglycemia. Basal insulin means a steady state insulin level that remains in our blood plasma, and that is also sometimes known as background insulin. So that is the level of insulin that is secreted from pancreas, and always some residual insulin, steady state value that remains in our blood is the basal insulin. Bolus, you might have heard of bolus insulin injections, means you might have seen some diabetic patient when they go for marriage parties, they know today they are going to take a lot of food and sweets. So what they will do is, just before the meal, they will calculate how much they are going to eat, and then accordingly they will take some insulin injection in their stomach. So they know that by the time insulin is in the body and they take the food, that insulin will utilize the glucose and their glucose level will come down. So that amount of injection that compensates directly for the glucose in the blood plasma is what is called bolus injections. Basal plasma glucose means the steady state value, the equilibrium value of the glucose level in the blood that remains always in a body average. If you do HbA1c test, you might have, some of you might have done those blood tests. HbA1c average, what is the steady state value that remains in our body? So that is what is the basal plasma glucose level. Okay, so that also gives us an indication of diabetes diagnosis. And this basal plasma level and basal insulin level of our body is not same for all people. It changes from person to person, and body size, what is your activity, stage of growth, hormone levels. Many clinical tests can be done to determine the level of basal plasma glucose and basal plasma insulin level of any patient under study. Okay, so doing through some clinical test, it is very easy to find out. Then there are two types of glucose tests, or you can say simply diabetic tests are done. And these diabetic tests are called glucose tolerance test biomedically. In biomedical engineering or in medical terminology, these are called GTT, glucose tolerance test. Means the ability of our body to utilize the glucose is what is the meaning of tolerance. And this can be measured using the tolerance test. Okay, so there are two types of tests that can be done. One is called oral glucose and another is intravenous glucose. Oral glucose test means we are asked to take orally some glucose and then we do the test. You might have done those random tests, postprandial test, fasting blood sugar test. So these are all part of our oral GTT. So in oral GTT, actually we are given some sugar solution. We eat some sugar solution orally, and that from the intestine it slowly diffuses into our blood, this glucose, and then we do some measurement of the glucose profiling, that how the glucose is rising and then falling, the profile we do the test. So that is OGTT. And intravenous glucose tests are for those patients who are admitted in ICU. ICU patients who cannot take food orally, but the glucose injection is given directly intravenously into their veins, and then the profile of glucose and insulin is measured. So this is intravenous GTT. So this is somehow the oral glucose tolerance test clinical procedure. You must have a overnight fasting minimum 8 hours, then blood urine sample collected during fasting to measure the initial condition of insulin and glucose. Then orally we have to take some glucose water, and then the samples will be collected every 30 minutes for glucose and insulin. And this is the glucose tolerance curve. You can see there are many curves. I will not discuss all, but yes, you can see the green curve. Green curve is always above some normal. Let me tell you, the normal level of glucose is 70 milligram per deciliter to 100 milligram per deciliter is the normal range. Okay, somebody may have glucose level 80, 90, 100, 110, 7. So this is normal range. So normal range is around this 70 to 100. So if some patient has got this type of curve from the test, then it is severe diabetic, because in fasting only it has got a high blood sugar, and after that a patient takes a blood glucose, the glucose it consumes, so glucose rises, but the fall of the glucose level in the blood is very low. It never comes to the normal position, so it is severe diabetic patient. And the red one you can see, in fasting glucose is normal, then the patient takes the glucose water, so it rises, and after 1 hour the glucose trend is coming down, and it remains. So this is the normal. Red one is the normal diabetic patient. It's not a diabetic patient, healthy patient. And the moderate one is, you can see in the fasting period only, it has little bit higher blood glucose. After that, when patient takes some food or glucose, it rises very rapidly. The slope of the curve is very steep. After that, the fall is very less. The slope of the curve is lower, and the level of the glucose tends to come to normal but does not comes back to the normal. So this is a normal patient graph from the test. We can find out this is moderate and this is severe. So this is how we interpret from profiling and clinical test who is diabetic, who is non-diabetic, who is pre-diabetic, and all that. So IVGGT means intravenous test. I've already said for ICU patient having malabsorptions or something like that. So here actually the glucose will be directly going to the veins, and blood samples are taken every 10 minutes for checking glucose level and insulin level. Okay, so that way this profiling will be done and diagnosis will be done for the patient who are not able to take glucose orally. And these are certain ranges for diagnosis of diabetes. If normal ranges for test scenario, fasting blood sugar should be within this range, which is normal. Random should be less than 140, and post-prandial means 2 hours after the food, if you test. So 2 hours or 1 hour after the food, if you test, it should be around 140 milligram per deciliter. Then it is normally a normal range, or the patient is healthy. And it is very important for us as an engineer also to know the dimensions. So international units that are followed for measuring the concentration level of glucose and insulin in a body. So for the glucose, it is molar concentration measured in millimole per liter. In United States, continental Europe, normally it is milligram per deciliter being followed. And we can always do a conversion from these two units, millimole to liter or milligram per deciliter, by some factors of divided and multiply by 8 either way. In India, you will find normally blood sugars are given in unit mg per deciliter by all the standard clinical practices. And this is diabetes diagnosis chart. I'm not going into it, but this diagnosis will vary from country to country, region to region within a country and across the world. There are many reasons for that. So these charts keeps on changing. It is just a guidelines. So suppose in India, people know that what should be our normal and diabetic ranges. In Europe, it is different because their race is different, food habit is different, their working style is different. So they have different, a little bit changes in the range, but not vast. Now let us come to try to understand what is an artificial pancreas. So in artificial pancreas, it is a technology under development to help people with type 1 diabetes. So let us be very clear. It is not a treatment or solution to type 2, but it is a solution to type 1 diabetes, such that we can have automatic control and continuous control of blood sugar by providing substitute endocrine functionality of a healthy pancreas. Because for type 1 diabetic patient, pancreas is completely not working, but with the help of some devices, we need to give the automatic functionality and continuous control of blood sugar as our body does in a healthy human being. So exactly same thing need to be replicated using electronic devices, and therefore it is known as artificial pancreas system. Let me tell you very quickly. If you can see the pointer, then this one is the MEMS-based sensor, which is called continuous glucose monitoring. So this is placed just underneath the skin, which is called subcutis or subcutaneously. So these are now very small devices are also coming. So this is an area where MEMS people can work on developing very accurate sensor for sensing the blood glucose level subcutaneously. So this is the sensor, and along with the sensor there will be some communication device. That communication device will sense wirelessly the level of glucose after every 15 minutes to this device. And this device, based on the level of the glucose measured after every 15 minutes, will excite the control algorithm to decide how much amount of insulin need to be injected into the body. So in this device, there is one insulin tank and a control algorithm. So this from this device, actually a command is being sent to an insulin pump to inject certain amount of insulin into the body through this catheter and cannula that is also fixed on the subcutaneous or underneath the skin of the patient. So this is again a MEMS device. So there are two contribution of people on artificial pancreas system is to design a very accurate MEMS infusion pump and the MEMS glucose monitoring devices. And these two devices has to work with the control algorithm very, very accurately. Because finally, the control algorithm is the brainchild, decision maker of injecting a right amount of insulin into the body. If the insulin is not rightly injected, then the metabolism or there may be a catastrophic complication in a body. So I believe that these are the two areas where people who are attending this ATAL FDP can try to find out that how to come up with these two MEMS devices, because it is not only to design device, also you need to design a software, okay, so that these devices should be integrated properly with the control algorithm and appropriate software such that the coordination between these three is done seamlessly and very accurately. So these are the engineering components of artificial pancreas system. These actuator are MEMS-based actuator actually. This actuator and sensors. And the control algorithm, actually, this is an embedded hardware or software which we need to design as a control engineer. And these are the area where the people from electronics and MEMS and mechanical people can work together. And the system is basically the patient, which who needs the control of glucose. So these are the engineering component. So this is how insulin pump or actuator looks like. This is the insulin infusion pump with the catheter, tank, and algorithms in this setup. There is a reservoir. This is the reservoir. There is a pump here, cannula, and catheter. So this is the manual operation that how with syringe actually we have been taking the thing. This is the traditional way. And this is how the sensor is connected in our body, as I've shown. So this is the skin. Just beneath the skin, we dip the thing. This is our blood capillary or blood vessel. So there is a diffusion of information of glucose from here to the interstitial fluid. The cell around the tissues actually has the information of glucose. So from there, actually, we pick up the level of glucose in our blood vessel. But there is some problem, let me tell you. The glucose level in the blood is important to measure. But if I have to measure the level of glucose from the blood vessel, then every time I have to prick some part of the body to get the blood, and blood measurement has to be done. That is time taking actually. And that is also hurting us very much. So people are trying to devise a BioMEMS or MEMS devices just can be placed just beneath the skin, and from there we can collect the information of glucose level from the blood. But the problem is, whatever is the level of glucose in the blood capillary and what is the level of glucose in the interstitial fluid, they will be different. Suppose if I take food, the level of glucose in the blood vessel will appear maybe within 30 minutes. But the level of exact value of glucose to appear in the skin or below the skin will have some time delay. There will be some time delay. So we need to consider that what is the amount of delay that we will have in the level of blood glucose to appear under the skin and the level of glucose in the blood vessel. So while designing our sensors, we need to do all this calculation and proper calibration by doing series of tests. This is the reason why these are very, very costly sensors, because a lot of money is spent on experimental data collection before the device is calibrated properly. So this is what is the story, and this is a manual operation of the glucose sensor prick type. Okay, but that hurts quite a lot. This does not hurt because it is a very small device, very small micro type of device that is installed just above the skin with this electrode just going a few micrometer inside your body. So it's not very painful. Okay. So this software means the computation algorithm and interconnection and communication devices between these three gives you the algorithm, computation algorithm and software part of it. So if I try to sum up what I have said so far is something I can visualize this entire artificial pancreas from the control perspective. Closed loop control system. This is my patient. This is the infusion pump. This is the feedback of the glucose from the body, which is the sensor or continuous glucose monitoring device. And then I have to maintain my glucose at a steady state. So this is the reference value. I will compare it with the reference value. Then there will be some control algorithm that will tell me that how much amount of insulin I must inject. And based on that command, insulin pump will inject that much of insulin into the patient body, no matter what is the disturbance I am giving to the body in terms of taking meal, food, exercise, no matter whatever I do. But as the system is completely closed loop, I need not worry about how much food I am taking, what food I am doing, no matter I don't have a pancreas, but this is completely an automated biological system where this is only biological. This is my actuators and sensors, which are engineering devices. Control algorithm is computation part, and then we can have this type of topology and we can try developing an artificial pancreas. Okay, so though it sounds quite theoretical, but yes, this is the reality actually, and the solution can be given for type 1 diabetic patient. So I'm not going into this. So may I ask Preeti that how much time do I have? It is almost 4 o'clock.
Yeah. Now you have some time. Okay, you can go on. No problem, because you are the only speaker in this session. So if you want to go further, you can go on.
Maybe I have 20-30 minutes.
Yes.
Okay. So I am not going into all these characteristic of insulin action and injections and all this, but let me tell you what is the Indian research scenario on artificial pancreas. That may be very interesting for some of the researchers. There is only one project so far being sanctioned by Ministry of Science and Technology on artificial pancreas, that has gone to Indian Institute of Science Bangalore in 2019 under Project Imprint. And it is my fortune that I worked with a person in 2013 to get myself trained on artificial pancreas system, who is now working and developing this. And with MS Ramaiah Hospital, Bangalore, clinical trials are on. They started in 2019 and 2021. They have started clinical trials of control and some MEMS devices that has been manufactured in CDEC ISC Bangalore Center. So sensors, I think they did not make, they have got it from market, but the pump, MEMS-based pump they have manufactured. The hardest part was to integrate two different topologies of the devices in terms of communicating between the indigenous pump which they have manufactured and the pump and the sensors they have bought from some other company. So they had a tough time actually integrating in terms of software or communication between these devices. That was they had a hardest time, and of course the control algorithm that they designed was not working properly for MEMS device. So let me tell you, there are a lot of challenges in control to control a very micro mechanical devices. I can control a big motor, no problem, because I can inject any amount of current and voltage and any gain I can calculate and give it. But if I have a very micro level electronics and mechanical devices, the level of gain that I need to calculate using control algorithm should be very judiciously, optimally calculated, because if the gain level becomes very high, there are chances that my MEMS device may be completely failing, right? So there are a lot of research challenges which has not been focused clearly, that how to design control for MEMS devices. There should be a high gain controller or there should be a low gain controller. What are the issues with the MEMS devices? These are something still an open area for artificial pancreas. And I know that I have been shared, as I worked with this research group long ago, some of the clinical trial results have been shared with me, and I have seen last week that there was a complete failure of the MEMS infusion pump and non-responsive behavior of the sensors, thus making the system again open loop, and therefore the control was not properly implemented. So the reasons we are still trying to understand, that the device was manufactured, but what happened, that why every time in clinical trial either sensor is becoming faulty, not giving communication, or sometimes the injection, that infusion pump is not taking the control command properly. If the control command has been to inject 5 units of insulin, the pump is injecting around 2.5 units of insulin. So there are a lot of problems and issues that have been observed for integration of control with these MEMS devices. So Indian scenario, we have said, is very bad. Only one project, that too in a laboratory prototype, is going on. Ethical clearance to clinical trials, it's only for laboratory purpose that has been given, not for any technology development or something. So I feel a lot of work here also we can try. To develop on our own and see a lot of testings we can do. But definitely we need a lot of data from medical field again to test those devices. So I don't know how to do, but yes, I feel there are a lot of ways to do it. Hardware in the loop experiment or in vitro some sort of experiment, if we can make a project and work on this direction. So APS, artificial pancreas system, are realized in 3 environments. One is called in silico, in vivo, and in vitro. In silico means all the components are mathematical models. This is a model. This is also a pump, is also a model. Device is also a model. Control algorithm is a model itself. So if everything is a mathematical model and we are doing simulation, but integrating it to give a realistic picture, then it is called in silico environment for clinical trials. And in vitro means in vitro in artificial pancreas system means here we can have a mouse or pig or whatever, some other animal, and all these can be our real devices, and we can test it whether our control algorithms, pump and devices are working or not. Okay, so we do not require too much of very rigorous ethical clearance. We require ethical clearance, but we can assume some animal trials here, and then we can try building this artificial pancreas and test our device APDs, artificial pancreas devices and artificial pancreas algorithms on this environment. So that can be called as in vitro environment. Other environment can be in vivo. When I say in vivo, that is the last stage of research. Now, I am happy with my pump. I am happy with my device. I am happy with my control algorithm. Now, this patient is a real human patient. And then we start the clinical trial. That becomes the in vivo environment. So, as far as I am concerned, I am still here because I don't have a group so that I can go into the biological component and extend into in vitro and in vivo. So I am still working in this and trying to test my algorithm for realization of APS. But yes, there are some good news because now modeling simulation is also taking a place where almost a realistic situation can be modeled, and then clinical, so many clinical scenario can be tested. So definitely now we come to a portion where it is called understanding the mathematical model for the patient, because if I can model the mathematical patients, then I can have the model of pump, or I can have the model of sensor algorithm and can do certain simulated model for testing my control algorithms. So there are many types of models: empirical model, physiological models, and other control oriented models from these test data. So, so far the physiological models are something which are very interesting because you understand the physiology and then try to create mathematical models. Some of the models which are in use are, these are the names, but the most common one is this one, which is called Bargman-Millimel model. Based on this, most of the work has been progressed, and these two are the most I can say the significant model that has been discovered in last 30 years. And now USA, FDA, Federation of Drug and something Administration, USA, has cleared these two mathematical model as more accurate model for doing some clinical studies and trials. And the model that is available, in silico model, those are available, are based on these two models, Overka and the Lamen. These are the two models. So let me now say that what are the challenges we have faced while designing control using modeling. The complexity of the metabolism between glucose and insulin. Yeah, there was some comments. Skip the slide. Which one? Which slide are you talking about? I cannot see the screen. Somebody talked about skipping slide there. Kaushik, can I take that question very quickly?
No, there is no such question actually. So on your slide, some some shaded portion is coming, no? That's why in the right hand side and this.
Yeah, right hand side there it is written that something I have skipped, so I could not read. It went away actually.
No, there is no such issue or no such question. So you can continue.
Okay, fine. Thank you. So challenges in control oriented modeling is that the entire body mechanism and metabolism, human metabolism, is really very complex. So this complexity, that human complexity and metabolism complexity, to bring everything in a mathematics is really a complex domain. And if we want to add each and everything, biological things, into a mathematical model, the model will become very complex. So if model becomes very complex, computation is going to be very complex. So definitely we have to do certain trade-off and capture only very specific and significant biological phenomena in the mathematical modeling. So of course, when we do modeling, we do little compromise. We have to do a compromise because capturing everything in mathematics means making computation very complex. So complexities are nonlinear functions. The parameters are not very straightforward. They are transcendental, discontinuity, these are very difficult mathematical function that's need to be handled in control algorithm. Then the parameters of the model are uncertain. So that needs to be handled because the body parameter cannot be very specific. From person to person, body parameter may change. With time also our body parameter changes. So all these things need to be addressed inside the mathematical model. Then the nature of the disturbances that comes from the devices. Okay, that also we need to understand. I have already said the behavior and the characteristic of the MEMS device is not well understood and taken into consideration when we decide or design a control algorithm. The characteristic of the devices, the nature of the devices are also not taken into consideration very appropriately. So there are a lot of complexities. There are a lot of scopes to for many people working on several disciplines to come together and to make it a reality. And also, we cannot make our size of the model very large, because if the size of the model becomes very, very large, then definitely the computation will become a big problem. So this Bargman minimal model is based on the data that is obtained from intravenous glucose tolerance test. And this is almost around many years now that this model people are working on. We also worked. And this is how the mathematical model comes. So I will quickly go back to this presentation. If you can see, I don't know, can you see my PPT now? I am showing PPT now, means Microsoft PPT, not the PDF. Can you see it?
So we are getting it in a shaded way.
No, right? So I need to share it again, I think. Okay, let me see. Okay, I think now you will be able to share it.
Yes, sir.
Yeah. So let us see how we derive this mathematical model. Now you can see it, I think. Yeah.
Yes, sir.
Now you can see that in doing the mathematical modeling for the patient in biomedical engineering, what we do is we have an approach is called compartmental model. We assume every body part to be a compartment, and the exchange of hormones takes place between a compartment. Suppose there are fluids and there are two chambers. Fluid flow between those compartments. The concentration of the fluid changes when there is a fluid flow and the reactions, right? So we adopt the same approach. We assume the body to be consisting of compartments, compartment of glucose, compartment of insulin, and then we try to see how the concentration is changing in the compartments. Okay, so you can see that I have shown here one red color compartment, which I am assuming that it is the concentration of glucose in this compartment. Blue color, I have assumed a compartment of insulin in our body, in the blood. Okay, and this is another compartment, the white color, which is the compartment of remote insulin, means the concentration of insulin in the interstitial tissues. Means just beneath our skin we have a lot of tissues and cells, right? So the concentration of insulin in those part of the body. So the body part has been divided into various compartments, and then we try to understand how it works. So these are some parameters. Meaning of the parameters: G of T, function of time, means with time how the glucose level or concentration is changing. Active insulin means I2 of T, which means the insulin concentration with time that is changing in our tissues, interstitial tissues and the fluid. I of T means the concentration of insulin in the blood with time how it is changing. And X of T is another variable that has been used to understand the delay of the effect of insulin onto the glucose. How much time it takes to act or react with the glucose. That delay is measured in terms of another variable. GB, it is the basal blood level. IB is the basal insulin level. I talked about basal values. VG, these are the volumes of volume of glucose, insulin in different compartments. and these are the flow rate, because there is a flow of glucose and insulin from blood plasma to the interstitial tissue. They are reacting. Some is going to the liver, some is going to the glucose is being also used by brain, by tissues, by liver, by kidney, right? So they are flowing out. They are coming to the blood. They are being distributed throughout the body. So there is flow rate. There is flow of this glucose and insulin throughout our body. So these are the flow rates with different units. These are the amount of effects that glucose and insulin have on each other. So I will not go too much in details. Maybe you are also not interested, and time is also not that much there. So we divide this entire derivation into two parts. First is glucose kinetics. So you see, this is glucose compartment. This is the remote insulin compartment. So what we do, we use mass balance equation. So this is very common in science. We have done that. So using the mass balance equation, we find out how much accumulated glucose we have in our blood vessels. So that is found out by the mass at the final time and mass at the initial time. So final mass minus initial mass is the accumulated mass or the basal value of mass in our blood. So what are the outgoing masses from the compartment? You see, there are two outgoing masses, and the outgoing masses are called the glucose uptake, means the utilization of the glucose. So these are, there are, when the glucose flows into the glucose compartment, of course, the when we eat food, glucose is produced, it comes to the compartment, then glucose flows out of the compartment. These are called uptakes, and also glucose is supplied to the compartment by the liver also. Okay, so there are two types of outgoing masses: glucose uptake by the liver, used up by the liver, and by the periphery. Periphery means tissue, brain, many other organs that is being used up. Okay. Other than external mass of glucose, there is incoming mass of glucose due to the production of glucose by the liver. So you can see an arrow coming in. So glucose is also produced by the liver. So the equilibrium or homeostasis says that if the concentration of glucose is greater than the basal value, then there will be the glucose uptake. The glucose will be utilized. If the concentration of glucose in your blood is less than the basal value or the threshold value of your body, then there will be production of glucose by the liver. So these are the two very important things for maintaining the equilibrium. The balance between glucose production and uptake. This is uptake and this is production. So if you have to reach to an equilibrium, this is what called NHGB, net hepatic glucose balance. So that balance need to be maintained. Whatever you produce, whatever is the incoming, in and out should be, a balance should be maintained, and that is what is called NHGB. We understand we need to have an equilibrium. The process of glucose uptake by peripherals, the tissue will be definitely using the glucose, the liver will be utilizing the glucose. So whatever net production, net utilization, so what you get is also affected by the concentration of insulin. Now you see, I have shown some arrow that this uptake, this utilization of the glucose is affected by the insulin, right? We have talked about it. The body secretes insulin, and this insulin reacts with the body in such a way the glucose is being utilized by the body. So if insulin is not present in a body, then glucose cannot be utilized by the liver, by the tissues. So your tissues will not get proper energy and glucose. So you will always feel weak, and body will not get energy. So insulin, effect of insulin for uptake and production is affected by this active insulin in the interstitial fluids. And this presence of insulin in the remote compartment is what we call it as a in the interstitial fluids, which we call it as a remote compartment, right? And this is the active insulin. So please remember one thing, that insulin in the blood does not react directly with the glucose in the blood. Insulin in the blood comes to the interstitial fluid. From there, it will react with the glucose in the blood vessels. So it is not directly from here. It is so first insulin has to flow into the remote compartment, and there is some amount of delay for the flow of insulin from here to here and then to react onto the glucose. So there is definitely some amount of delay, and that delay need to be modeled. So now I'm not going into mathematics. So I'm doing some balancing equations and putting the dynamics. These are all mass equations. I hope you understand volume into, these are the volumes and I think I have missed out something, volume into the concentration, that will give you the mass. So these are also mass, flow into concentration into time. So this will give me the mass. So this is a mass balance equation. So with balancing the mass and all that, I will finally get some differential equation, the dynamics between glucose and insulin. So that comes very logically from mass balance equation of physics, and from there we can come and arrive at a differential equation. This is directly from physiology understanding, right? So finally, you see, I think, yeah. So now this is the dynamics of the glucose with respect to time. Now, as I have said that there exists some amount of delay to act between glucose and insulin, because insulin appears in the remote compartment from the blood. So there is some amount of delay that needs to be modeled. So again, using mass balance equation between these two compartments, we can do the differential equation modeling. And finally, you see that I get two differential equation. This I already got. I captured the delay in the system, and then I got two differential equation. This x of t is nothing but the delay effect of insulin to react on t. That's all. Okay. And I am still important to tell you two things, that two important things that to be remembered in glucose insulin metabolism is what is the effectiveness of glucose and what is insulin sensitivity. These are the two very important parameters in modeling that needs to be captured. Glucose effectiveness means it is the insulin independent glucose uptake rate, means the utilization of glucose in the body not dependent on the effect of insulin is called glucose effectiveness of our body. Insulin sensitivity means ability of the insulin to enhance the effectiveness of the glucose, means with the help of insulin, how sensitive is the insulin to react with the glucose such that glucose can be utilized properly in the body. Because if glucose is not utilized, it means glucose level cannot be controlled. So using these two concepts, now you see that I got into, I modified these two concepts. I modified the equation of this delay differential equation. This delay effect I have modified because I have redefined my x of t in terms of some parameters. And finally, I have also modified my, these are simple mathematical flow that using this physiological understanding, we could modify this dynamics of the glucose and the delay effect. And finally, you see, I have added a parameter into the model. Original model does not have this parameter, but this parameter has been added extra here. D of t, it is nothing, it is diet or the meal disturbance, the food that we take. So when we take the food, initially the effect is very high, and slowly the food effect will decrease, right? The effect of the food will decrease, or the rate of appearance of glucose will decrease. So this is called Fisher model of the meal that has been included here. So glucose appears in the body because of the food or the meal that is also added into this differential equation. Okay. So instead of adding d of t and using this static equation, we have actually made this equation in the form of a differential equation. So this curve, nature of the curve for meal has been written in terms of a dynamic equation, and we got these three equations now. One is glucose concentration, delay in the effect, and this is the meal behavior that we take. Okay. Next is we need to understand the kinetics of the insulin. Now you see, this is the pancreas, this is glucose level. I have already said that insulin in the blood will only be secreted only when the level of the blood glucose will rise. Then there will be biosensor will give some signal to the pancreas. Pancreas will produce the insulin and put it into the blood. So that needs to be captured now in the differential equation. So again, using mass balance equation, we have tried to capture this particular phenomena. And finally, we got the dynamics of insulin into our body. Okay, into our model. Sorry. So the parameters, I am not going into it, just a rough idea how do we do the modeling. So now you see we have 4 differential equations, one for the glucose, one for the insulin, one for the delay effect of insulin onto the glucose, and one to bring the effect of meal into the glucose level. So now you can see this is the model for healthy patient or type 2 diabetic. This is for type 1 diabetic. What is the difference? Let us find. All the models are same, except you see in the last differential equation, this term. This is missing here for type 1 diabetic patient. So what have we done? This term is nothing, but this is the pancreas function. Pancreas works in such a way that if this threshold level P5 is greater than, less than, or something, then based on that, there is signaling from pancreas beta cell to secrete insulin, and then the dynamics of the insulin increases. So it happens in a healthy patient that insulin is secreted automatically by the body, and this function depicts the automatic release of insulin into the blood. But for type 1 diabetic patient, as pancreas itself is not present, so this function tends to 0. So what happens in that case? We need to inject insulin from external source. So this is how we need to design our control U of T. So we need to have a control algorithm to calculate that what should be the appropriate dose of insulin that has to be injected externally for the type 1 diabetic patient. So this is what all about the modeling concept in short. So let me go back to content. Okay, I hope now you can see my other picture.
Yes, sir.
Yeah. So this is all about the modeling portion. I'm not going much details now. I don't have time. So we have created a model and we have shown an open loop response. Open loop response means when we don't have a sensor data under open loop condition, we have given some meal disturbance, means we have taken some amount of glucose in our body, not the food, not the carbohydrate, but carbohydrate has been converted into glucose, and that glucose amount we have injected into the body. Now we see what happens to the glucose profile, right? It starts from 200 milligram per deciliter initial value, give some glucose injection. So what happens? The glucose rises and again tries to fall. Again, we give some dose of glucose. It again rises and start to fall. But you always see that it does not come back to normal position. So it means this simulation has been done for a type 1 diabetic patient. It does not come back to the normal level. And when we eat food, the glucose level rises. At the same time, we have calculated how much insulin need to be injected. Manually, we have calculated. We have given some doses of insulin. Then we saw that the rates are decreasing. If we give less amount of insulin, maybe the rate of decay of glucose will be lesser. So we tried to study the profile of glucose and insulin and how the reaction takes place in the model. So these are some models that are much more advanced models or subcutaneous patient models. So in subcutaneous patient model, you see insulin, in this model, insulin is given directly to the veins, intravenous, because these models are coming from IVGGT models. But subcutaneous means insulin injection is given just beneath the skin. So from the skin, it goes to the interstitial fluid, and from the fluid, it diffuses onto the blood plasma. So there is lot amount of delay in the insulin, and the distribution of the insulin is also not done in one single compartment, but it is done in different compartments. Okay, so more detailed model. I'm not going into it. There are many such models, but finally we proposed some model, because this model, what I'm talking about, there are maybe around 17-18 equations, but 17-18 equations to handle in a computation is tough. So we tried to come up with some new model of the patient, where we have only 7 to 8 number of differential equations to solve. And this model performance definitely has given us a good results. So but now what happens, you see, we have tried to club up the physiology from different papers, and then we came up with a model. So definitely there was a need of parameter estimation. We need to estimate the model parameters. So we did some sort of nonlinear least square fitting of parameter estimation. And after doing some parameter estimation, we had certain patient data also available. So with the patient data, and we tried to fit our model. So you can see the red one is simulated, and this is the patient data. So our model tries to fit quite accurately with the patient data that we have, and that was used for parameter estimation. So these are certain statistical calculation of the parameters that we have, coefficient of determination that we did with certain data and the model. So that was to some extent not accurate, but definitely significant, I will say. So this is the control problem, which I have already talked about. Control challenges I have talked about. I'm not going into the control derivations. These are some control mathematics. You might not be liking it, but I will show you the result of our proposed control strategy on the models. So you see, this is a grid analysis where we have done certain grid analysis. I was talking of hypoglycemia. Our control algorithm should be such that the decision that we take to inject insulin into the patient body should not be such that hypoglycemic condition is created. Because if hypoglycemic condition is created, then there will be a patient death, or patient may go to coma or seizures, short-term complications may be there. So we need to design our control in such a way and prove that the decision of injecting the insulin avoids hypoglycemic condition. So you can see the maximum plasma glucose concentration on y-axis and minimum plasma glucose concentration on x-axis. And you see our control algorithm says that at all level of time, because of the insulin injection from our controller, right, keeps the glucose concentration within 180 se jada, and minimum it is around 180 or something like that. So we just wanted to show that hypoglycemic condition was avoided somehow in this control algorithm, using this control algorithm. Wherever I want to say that in this case, we also assume that patient parameter is varying. Plus minus 30% random variation of parameter was also taken for the patient. But still, we showed that hypoglycemic condition is completely avoided. Means we do not aggressively give insulin injection into the body. Still, we try to control. So there was another strategy of control. And in this strategy of control, again, we have shown that our glucose level is within this range. Again, a parametric variability of model was plus minus 20% randomly. This is another adaptive control framework that we designed. I'm not going into it. Now you see again the ranges of level of glucose that has been controlled is around this range. So it should not fall below 70, right? 70 is the normal range, right? Or less than 50, it should not be. So nowhere our minimum level of glucose has gone beyond less than 50 or 60. So we tried to show that hypoglycemic condition using our control algorithm is completely avoided. So this is in insulin pump modeling that we have done. And this is of significance for MEMS people because they may require these insulin pump models for designing the actuators. So we did some modeling on this. We find out some transfer function. We did work on model of glucose monitoring little bit to identify what type of noise does the sensors gives. So some noise modeling was done. And we found that the response of the noise and whatever model we created a little bit agrees with the characteristic of the CGM devices. And then we tried to make one in silico complete artificial pancreas. So there was device model, pump model, control algorithm, patient model, and then we tried to simulate this entire scenario and we got some results, but they are not very promising. We're still working on integrated model of device and patients together. So this is something for you. MEMS devices, I have just given a reference people are working, which is called APD, Artificial Pancreas Device Systems. So people are trying for thin film insulin pumps. This dome shaped thing is for pressure to be created for pumping of the insulin accurately. So this is called nitinol MEMS pump. And I think this they have patented also, and but it's not commercialized. And this is another artificial pancreas pump architecture that has been designed, and this is commercial product already available. This is called D-Biotech some pump system. So these are some of the features of this MEMS pump. You have multiple sensors to deliver single control 200 nanoliters each units, 0.02 units of insulin it can deliver. Sensors can detect malfunction and occultation. Means if you have the blockade in your cannula, catheter, and if the pumping is not taking properly, these devices have there are sensors to detect occultations and malfunction within few strokes of pumping. Because of MEMS size integration, very small, such pump can be filled up to 5 ml of insulin for continuous 7-day use, which is also very important. Because every time if I have to change my insulin pump, that is also not advisable. And these pumps and MEMS devices are better suited for cyber physical framework of artificial pancreas system. So I think I have finished. I will just play a video which is maybe 2 minutes long, just to give you a motivation that the things are not hypothetical, but I don't know what has happened again. Yeah. So this is another technology towards MEMS devices to show that there are 3 generations in progress for artificial pancreas devices research. It started with first generation, then it went on to the second generation, and now we are looking at fully automated thing where the MEMS devices will play a greater role to have a fully automated. Now also fully automated devices are not available. People have got a little bit of hybrid. Some of the things are automatic, some of the things are still manual. But the research is going on how to make it 3rd generation artificial pancreas with full automation, full accuracy, and full alarm and sensoring system that will completely take away doctors from the loop. Actually, still in artificial pancreas system, doctors are hooked into the loop. But people are now looking into this 3rd generation development. And for 3rd generation development, it is very, very necessary that device people and control people should work hand in hand, because let me see. Yeah. APD system has to work with control and fault detection algorithms. So careful selection of control algorithm for these devices are very, very important to arrive at a third generation artificial pancreas system. The Indian technology that is going on, it is still in first generation artificial pancreas laboratory prototype, and third generation we have still not thought about because fault detection is still not taking place there. So there can be three ways by which MEMS and control can be integrated. I have given here. First, your control function could be integrated into the MEMS device with smartphone or smartwatch app, providing the user interface with controller. Second can be control function could be performed by a separate mobile controller device connected wireless to CGM and pump. And the third can be in the form of app installed on the regular smartphone or smartwatch. The APD control app would run along all other functions performed by the such technologies. So these are the three frameworks of control and device that needs to be integrated for coming up to completely automated third generation artificial pancreas system. So I think this was the only addition from my end for this workshop. I never worked on it, but I read for some 3-4 days to find a linkage of artificial pancreas with BioMEMS. Now let us see a video, and then it's time to say thank you and take some questions from you. Let me see where it is. I go to sharing content. Can you see this index C or something like this?
Yes, sir.
Yes, sir.
Okay, let me, I'm playing. Please tell, can you hear the music and the voice audio is clear?
The audio is not. We can't hear the audio.
Not audible, I think.
Yes, sir. The video is
Not
clear,
audible?
but we can't see.
Nah, sound
Yeah.
is not coming. Video we
Oh.
can see.
How to get the sound, I don't know. I have shared.
Saurabh, what is that mechanism to get the sound?
Sir, you please reshare your video by clicking on optimizing motion video. There is an option in sharing option is there for in WebEx motion video optimization.
Motion video optimization.
Yes, if so just
I have only share web browser. I will share web browser just.
okay.
Let me see.
Share web browser. There is an option.
The optimized video, na?
Yes, yes.
Optimized.
But I am getting only share content, share web browser, share my meeting window. That's all.
Oh, there is a there is a window in that window. In the right side panel, optimization is there. Optimization, you have to select the optimization, which type of optimization you are requiring. So in that you can select the motion video. How can you?
Acha. Okay. Optimize for motion and video, right?
Yes.
Select it.
Okay, I selected it, and then I can go to that Google browser and share it, right?
Yes. Now it will open.
Now it will. You see, can I do that? I think.
Here,
Tell me now, can you hear the sound?
Yes, sir. Sound is coming, but video is not coming.
When night falls in Kibbutz Ma'ale HaChamisha, 18 children go to sleep hooked up to laptops. This is the first trial of its kind. At night, their blood glucose levels will depend on these computers that function as an artificial pancreas.
Yes.
Coming.
Now
Okay.
it is fine.
Okay,
Fine.
just a minute.
When night falls in Kibbutz Ma'aleh HaChamisha, 18 children go to sleep, hooked up to laptops. This is the first trial of its kind. At night, their blood glucose levels will depend on these computers that function as an artificial pancreas.
דותן בן 11.5 ויש לי 7 שנים סכרת. כדי נשארת ואני בת 12 ויש לי 4.5 שנים סכרת.
Here, 18 children with diabetes get acquainted with one another.
יש לי 13 סכרת, אני לא זוכרת, ואני די רגיל לזה. תגיד את זה ככה.
בוא נעשה ככה, יש לי 7 שנה סכרת ואני די רגיל לזה. זהו, הם נותנים יותר מדי זמן לשיחות האלה. 5 דקות זה לא של חצי דקה.
Dotan Yam Sirkis has been sick as long as he can remember. He's an excellent tennis player and feels like an ordinary child. Almost.
3 מקצועות בחיים. אני לא יכול להיות לו טייס של מטוס נוסעים, מטוס פרטי אני יכול, ואני לא יכול להיות נהג אוטובוס או נהג מונית. זהו, חוץ מזה, לפי מה שאני יודע, אני יכול לעשות מה שאני רוצה.
True, but the diabetic's path to doing whatever they want is much longer than the path of healthy children. First, there are these tests. Every few hours, they must check their blood glucose level.
רמות סוכר בדם כשהן גבוהות מדי גורמות לסיבוכים ארוכי טווח, לפגיעות בעיניים, לפגיעה בכליות ולפגיעות בגפיים. כשהסוכר נמוך מדי, חולה הסוכרת חווה סיבוכים מיידיים. הוא מאבד את ההכרה, הוא יכול לפרכס, ואפילו זה מסכן חיים.
אתם לא מתחילים לאכול לפני שהמדריך ראה מה יש לכם בצלחת ורשם את מה שיש לכם בצלחת ושהרופא אישר לכם בולוס עליו.
Bolus in diabetic slang means another injection of insulin. In diabetics, the pancreas can't control the blood glucose levels. After a meal with carbohydrates, everyone's blood glucose rises. The diabetics must inject insulin to lower it. To gauge the amount of carbohydrates and how much insulin to inject, one must excel at math.
יש לך חישוב של כמה פחמימות זה כל לחמניה. 350 35 זה לחמניה, תני 10 גרם.
כל פעולה שאני עושה או אוכלת, אז אני צריכה לעשות את זה לפני, אני צריכה להזריק את האינסולין ואני צריכה למדוד. וזה כאילו חצי מהדברים שאני עושה, אז כזה לוקח חצי מהחיים.
The trial begins at 4 o'clock. The children enter a room with 18 laptops, and for the first time, they hook up to the artificial pancreas.
זה מרגיע. כן. שהניסוי מתחיל לעבוד.
הלבלב המלאכותי בא להחליף את הלבלב הפגוע של חולי הסוכרת.
How does it work? 2 devices are hooked up to the child's body. One is the insulin pump. The other is a sensor that monitors the blood glucose levels. The artificial pancreas should link these 2 devices by getting data of the glucose levels from the sensor and automatically instructing the pump how much insulin to inject.
וכמה תקוות אתה תולה בניסוי הזה?
אני מתאר לעצמי שזה יצליח. זה הכל עושה לבד. אם צריך זה מזריק, אם צריך זה מונע את ההזרקות, זה הכל לבד. אנחנו לא צריכים להתעסק בזה.
אתם יכולות לדמיין שוב חיים כאלה שאתם לא מתעסקות בזה יותר?
יהיה קצת קשה להיפרד. כן, יש בזה דברים טובים גם.
מה?
קודם כל זה יותר אחריות, אז זה יותר מלמד אותך איך להיות יותר אחראי.
From now on, the children will be connected to the laptops, to their artificial pancreases at all times, even when they watch a movie.
אנחנו רואים את ההחלטות שהלבלב הזה עושה. הוא החליט להזריק פה יחידה אחת, כשרמות הסוכר מתחילות לעלות הוא מעלה חזרה את קצב הזלפת האינסולין. כל ההחלטות האלה נעשות אוטומטית על ידי המחשב.
The system also conveys an alert when there's a problem. That's why Dr. Nimri is looking for Dotan. When Dr. Nimri finds Dotan, Eran, the trial engineer, is already with him.
החיישן בעצם איבד את המשאבה, אז אנחנו מנסים להחזיר את החיישן למשאבה, שהחיישן והמשאבה יחזרו לתפקיד ביחד.
The malfunction is repaired and Dotan goes to his room. A phone call from home.
אבא.
היי דותן, תגיד לי איך הסוכר שלך?
הסוכר די מאוזן.
מה קורה אם אתה תהיה באמצע לילה נמוך?
הכל עולה במחשב הגדול של הרופאים בלייב.
10:30 and it's lights out. 18 artificial pancreases are now put to the test.
דותן, בסדר, 120.
The doctors and engineers are alert in the control room.
זה 73 בסנסור, אבל החיישן שלה יותר גבוה. ניצן 76, קורא אותו זה 76.
הסוף של הפרויקט הזה זה ילדים מטופלי סכרת שלא חושבים על הסכרת שלהם. פשוט מאוד.
Yes, though Dotan, Eden, and all the other children taking part in the trial find the positive aspect, even in diabetes, they went to sleep hoping the trial will work. That one day they will wake up and no longer feel the disease known as diabetes.
So thank you very much. So that's all from my end. And it was basically an awareness to tell you that yes, the disease exists, but there is a need of engineers to make it completely a solution from technocrats and engineers to this problem. Thank you. Thank you very much. Any questions I can take now?
Thank you, Dr. Day. It was a wonderful the session, the good blend of audio and video and presentation, everything. So it was a nice experience that you have given a new and innovative domain for the research, specifically for the MEMS and microfluidics researchers. And also that we also believe that not only the design and development and simulation modeling is required, but ultimately whenever we invent the device, the full controlling of the device is very, very important. So that's why when with keeping in this mind, so I have thought that okay, I would invite Dr. Day. He's a very accomplished researcher in the control engineering that I know. So people will be immensely benefited out of your lecture. So how to involve the control system also to control all the medical implantable devices. So you have rightly given a good directions, and the hope that also the control people, electrical engineering people and electronics, basically all the people will come together and then we can do a very, very good and novel device for our the society or the common people. Yeah. So now with this, I think we can take very few questions, okay, because already we had a long session and very good experience on that. I think the channel is open for the participants, those who want to interact or ask some questions to our speaker. You can unmute yourself also, you can directly interact with our speakers. Please carry on. So any participants, if you want to ask something, you can unmute and ask our speaker.
Somebody have asked, tell me something about your simulation software. Once modeling completed, how to write a program for simulation software, which software is best? software we used is MATLAB and Simulink. I have told you that there is one model that has been cleared by FDA USA for clinical trials and to understand artificial pancreas is called UVA Padova model. University of Virginia and University of Padova in Italy, they actually jointly came out with this model, in silico model, which is written in MATLAB Simulink, and NIT Silchar has got that model. We purchased it, and there are 30 patient data of type 1 diabetes also available there, and it does not cost much. You can also purchase. It is $150 only. That gives you 30 patient data also, and everything is done in MATLAB Simulink. So no worry, all academicians are acquainted with MATLAB and Simulink very much. So that is an answer to this question. Any other question? If it is there, let me see. Somebody wrote, you skip one slide after APS control. Please explain. I don't know which slide. If you can unmute and can tell me, I can.
Yeah.
US already having marketed product of MEMS APS. Yes, very correct. They have already marketed, but the problem is the cost of artificial pancreas from US to India around 15 lakh rupees. So for our common people of developing country, it is very, very difficult to afford that. Plus the cost of insulin every week cannot be afforded. So therefore, India is trying to indigenously generate and create an artificial pancreas system at the cost of around 2 to 3 lakhs, where we can convince the policy makers of insurance company that if anybody is type 1 diabetic patient, then the cost of that should be included in the insurance, also medical insurance, which is at the moment not being covered under insurance policy in India because the cost is very, very high. So India is trying to come indigenously with the pump, with the sensor and the control algorithm and embedded hardware such that it becomes a reality at a low cost. And that is the practice that IISc Bangalore is trying, but definitely it's a lab type. I think more and more research, interdisciplinary research from device, control and embedded is required at this stage to get a low cost solution for artificial pancreas project. Yes, Medtronic, Dexcom, they are all available in India, but they are selling not their technology. We have tried and approached Medtronics. Medtronics does not give their software. They give you the device. So if they don't give the software, you don't know how to operate and integrate. So it is better to come up with our own technology, hardware and software that we can integrate. So you are very correct. US has already done it, but the technology is not known by Indians and the cost is very high. Simulation software I cannot explain, but if you write to me in my email, yes, I can send you the simulation guidelines to you. I don't think any other question.
Yes.
Yeah. So,
yes.
Dr. Kaushik, I think it is over.
Priti will conclude the session.
Excuse me, may I have one question from Rajiv? I'm sorry for being a bit late on asking the questions.
Yes. Go ahead, sir. Go ahead.
Hi, Dr. Rajiv. This is Rakesh. Rakesh from one of the government college in Jammu and Kashmir. I hope, you know, Jammu and Kashmir is a popular place whenever it comes around.
Sure. We know.
Thank you very much indeed. Nice presentation. I really like, you know, the way this program has been so far. This is my area of expert, and I miss a couple of days just because, you know, the problems in Jammu are different the problems in other part of India. So we do have a network issues, we do have a power cut and so many things. But the way this BioMEMS FDP has been going on, I would say that I have attended 4 in this year, and this is one of the topmost and the best one.
Thanks.
So besides that we got a wonderful presenters here, I might have a technical question because as somebody was asking, like, the clinical trials in India. Yes, they are difficult. It's very expensive, and then we do have a lot more population to put into any of the BioMEMS or based device or technology into the market. What I'm interested, I've been a worker and been a researcher on, I did work on carbon nanotube based cardiac markers detection and sort of like prediction. But the problem still is the specificity. Whatever in these technology-based solutions, we do say, like, we are getting a specificity to a percentage of that and this. But unfortunately, it's a bio world, and bio world is very, very complex. If I were to just look for a nitrogen oxide in the environment, I probably would use like silicon nitride based or silicon oxide based nanoparticles, and would say, like, yes, my senses is more sensitive to nitrogen oxide, but less sensitive to the NOx2 or NOx3. But in case of bio, I just want to know your comments, like, how we can look more on the specificity side and then predict the real and accurate data instead of what we do in labs or in just on a desktop sort of like experiments.
Okay, thank you, Rakesh. And I think your question is very much specific to the material science, devices, and that also having connection to bio world. So I am not an expert or someone to comment on that, but what I perceive is that the problem that you face in your domain of research and connecting it to bio application, exactly the similar type of problem we do face as a computational scientist, and finding out a solution which is quite practical and implementable and also convincing for the medical fraternity. Because when we talk about our model that we have created out of physiological understanding and then simulated in a desktop world, and the computational algorithm that we have designed, it becomes very difficult for us to make medical fraternity understand that this will work in a real world, number one. Second part is that the type of cohesiveness that we have or understanding we have between medical fraternity and engineers is not there. That's the reason why we don't sit down across a table and try to understand our problem together and resolve it. Right. This is the major reason. Second, I do not agree that all the things that we do, all the computation that we do will not work in reality. But yes, there is something that people do not have an acceptance limit. Medical fraternity do not accept exactly what do we propose. It's not that we do all rubbish in a desktop. So definitely, if we get a chance to test or create an environment, our computational algorithm, or from a device side or a material engineer, at least if you are given a chance to work, at least on a laboratory prototype, with taking all the precautions that you can take during testing, and that help has to come from medical fraternity to us, right? Then I think, of course, it's that at the first step, reading a paper and directly finding designing a technology is not a possibility. I can design you can design, or Dr. Kaushik can design a MEM device by looking at the principles, but will it work or not work? That has to be tested, and for that, the acceptance from medical fraternity is required. A framework is required where we get a chance to test our devices. So that is what is missing, and because of which I think we actually cannot propose such type of project. We cannot get such type of project, and we are lagging behind here. But it appears that we are doing all rubbish sitting in a desktop because we are not in a position to test it in a real environment. We are not given a chance to test it in a real environment. That can be actually my particular view. But given a chance to test something on a real environment, even on a animal trials also, if we can do, I think we can find a solution.
Yeah, right. Thank you, Dr. Rajiv. I think, you know, my point was never been underestimating the potential of software simulation of any experiment environments. I think nowadays, like, you know, whenever we go for any experimental study, we do have a simulation in advance, and it's playing a vital role. You know, it doesn't matter whether we are doing a material science or we are in a clean room doing device fabrication, unless we get like a 99.99% prediction from our simulation, then we do go for trying the same protocols and recipes that comes from the simulation. I think simulation is providing a pathway where we can look more into the reality or the success of any experiment results. So that was not a point. I'm very happy to, you know, have your comments on it, that the medical fraternity has to accept the simulations also before we start looking for generating the real environment for testing our you know, proposed technology. Thank you very much, Rajiv, and thank you very much, Dr. Kaushal.
Thank you very much. I think you should see the speaker also. So I will open the video for a short time, because I did not open my video because of bandwidth limitation and all that. So I don't know, will it open or not? You see, my bandwidth may not be very good. I think it is not coming. So again, a bandwidth issue. Anyway, fine. So I am done. Preeti, go ahead. You have some formality to do.
So thank you very much, sir. This is not just a formality, but it's indeed from the depth of our heart that we thank you this evening. Thank you for all this informative and detailed talk on artificial pancreas system and the role of MEMS, how MEMS can fit into it and really deliver a real-time working model. Thank you for the details on biomedical preliminaries, diabetes mellitus, artificial pancreas, and their broad research scope that you have explained to us, that there's a lot of scope yet to be done in the design of pancreas. Thank you for sharing the mathematical model concept, sir. And indeed, we are thankful for the motivational video that you have shown us. Like it always looks like the diabetes is a common problem. Like one elderly in each home is having diabetes. That's the only information that we common people know. But this type 1 diabetes and the children who are suffering with that, the girl in the video who said that half our life is taken off in calculating our insulin, is really like they live half of life is gone in that. Is a saddening thing, but technology is involving in that, and we are able to try to help them is a real encouraging thing, sir. And we really hope that we people from the technical field would be really able to relate with the medical problems and deliver real-time solutions to them. Thanks a lot for your talk, sir. This is really enlightening to us. Thank you very much.
Thank you, Dr. Dev. Thank you.
Dr. Kaushik, thank you for having me on board in this workshop, though I was really out of track, but definitely I somehow managed it. Thank you very much for having me on board.
You are not absolutely out of track, so that's why I told the control and these things are also required in this field. Without anything, so it cannot be a complete device and system. So control is required. Yeah. Thank you, sir.
Thank you very much. Thank you very much.
Sir.
Thank you. So thanks all the participants. For today we have closed. Then just I am announcing the tomorrow session. Tomorrow again we will start at 10 o'clock in the morning. Our first session will be also one hands-on training in some development of design and development of biomedical device. And it will be delivered by Dr. Sripada Rajake from Intelligence Private Limited. Then at 11:30 we have a second session also. It is also going to be very interesting and charming that the 3D printed microfluidic system for cell culture, and it will be delivered by Dr. Anil Kumar Piyar, who is a very senior scientist, very renowned scientist in the SCTI NST Kerala. Then in the afternoon, so at 2:30, we'll have one more good talk by Professor G. K. Ananta Suresh from IISc Bangalore. He'll be talking on grasping cells. So after that, as per the mandate of ATAL ICT, there will be one test conducted from 4 to 5 p.m. and then from 5 to 6 p.m. Obviously, we will again meet for the closing ceremony of the entire 5-day program. That is the valedictory session of the 5 days program. So this is the brief about the tomorrow's program. I mean, with the same enthusiasm and zeal and the same kind of motivation, you please cooperate with us tomorrow also. We join in the morning at 9:50 a.m. so that we can start our program at 10 o'clock if there is no such technical issue at the speaker end or at our end, because technical issue can happen at any time. So please bear with us if that same thing happens tomorrow, but definitely we can resolve it, and then we will try to present you also the good session tomorrow also. Thank you very much to all the participants. Have a good day and a good night also. So we'll come back again tomorrow morning at 10 o'clock. Thank you. Thank you very much.